Padmarao?
పద్మారావు గారు మీరే హోస్ట్ ఇప్పుడు. పద్మారావు గారు ఓకేనా? ఓకే మేడం మీరు స్టార్ట్ చేసేచ్చండి. ఎవ్రీథింగ్ ఓకే.
ఓకే సార్.
యూట్యూబ్ లైవ్ కూడా ఇచ్చేసాం.
ఓకే
స్టార్ట్ చేసేచ్చండి.
సార్ గుడ్ ఈవినింగ్ సార్. డాక్టర్ రఘునాథరావు గారు గుడ్ ఈవినింగ్ సార్. Sorry for the delay in starting the program.
Fine with me, madam. Thank you. Sir, good evening, madam. No problem.
Sir, we have been encountering with some network.
No problem, madam. It's all right. No problem at all. Please. No problem.
Sir. Thank you very much, sir. Thank you for your patience. Thank you.
I have my presentation also. If you want me to broadcast from here, I can share my screen, provided the webmaster gives me the control.
Yes, sir. Please, sir. Okay. If you can, please share.
I have the presentation on my laptop also. I can share, madam. After VC sir's talk is over, then only I will start. It won't look nice otherwise.
Right, sir. Right. Thank you, sir. Thank you. Sir, Jagan, sir. Start చేసేస్తారా?
మేడం
దీప్తి మేడం ఆగండి సార్ జాయిన్ అవ్వాలి.
అదే చెప్తాను. ఇంకా మీరు రెడీగా ఉన్నారంటే చెప్తాను.
వీసీ గారు నేను రెడీ. వీసీ గారు వస్తే
వీసీ గారు అయితే వచ్చారండి. ఆడియో వీడియో ఆఫ్ చేశారండి.
పద్మరావు నా వాయిస్ వినిపిస్తుంది?
Yes sir.
Srinivas, I am able to.
Yes, audible sir.
Audible. Okay. Thank you.
Jagan sir.
Hello.
Jagan sir.
Hello.
Hello, Dr. Deepti.
Yes, sir. Yes.
You can start.
Welcome, sir. Welcome, sir.
Sir, good afternoon.
Good afternoon. Good afternoon, Dr. Ramaneshwari.
Good afternoon, sir.
Good afternoon, Jagan Mohan Reddy. Good afternoon, Professor Ramesh and Webmaster Srinivas and others. Let us start.
Myself, Dr. B. Jagan Mohan Reddy, working as an assistant professor and head department of chemistry and also executive council member of Pathophysiology University. Welcome you all for today's international webinar on COVID-19, contemplation on its pathophysiology and possible ending. I am really privileged to have the pleasant task of welcoming to this international webinar among this distinguished gathering. Respected esteemed Vice Chancellor, a great visionary and eminent scholar, Professor Mokka Jagannatharao Garu, welcome you, sir. Respected Registrar, Professor B. Gangarao Garu, respected Principal of College of Science and Technology, Dr. K. Ramaneshwari Garu, respected Executive Council members, Professor K. S. Ramesh Garu, who is working as a Principal of College of Arts and Commerce, and Professor Ashok Garu, and all principals and officers of our university. Respected today's speakers of this international webinar, Professor Kuchimudi Suresh Verma, Penn State University, United States of America. Another speaker, Dr. D. Raghunath Rao Garu, Founder Director, Homi Baba Cancer Research Institute and Research Center, which is located in Visakhapatnam. Welcome you, sir. All teaching faculty members and research scholars of various colleges and universities of our affiliated colleges also. All students all over the nation. As you are aware that national lockdown has extended till June 30th. During this lockdown period of pandemic situation, I'm very happy to say that and proudly our esteemed Vice Chancellor sir has taken great initiation and efforts in conducting so many online national and international webinars like this in our university, in each and every department. We are very much thankful to our Vice Chancellor sir. With great pleasure, this is to inform you that our Department of Chemistry is organizing this international webinar, and I congratulate my colleague who is working as Assistant Professor in the Department of Chemistry, Dr. K. Deepti, for this occasion and for her efforts. With this, I am last Telugu quote. I am closing that welcoming today's international webinar. Pani nundi visrantiki, visranti nundi paniloki marutunadame ఉత్తమోత్తమైన కార్యం అన్న వివేకానంద స్వామి సూక్తిని అమలు పరుస్తున్న మన గొప్ప కార్యశీలి పరిపాలనా దక్షుడైన గౌరవనీయ వైస్ ఛాన్సలర్ శ్రీ మొక్క జగన్నాథరావు గారికి నమస్కారాలు తెలియజేసుకుంటూ ఈ కార్యక్రమానికి కన్వీనర్ గా వ్యవహరిస్తున్న డాక్టర్ కె దీప్తి గారిని ఓపెనింగ్ రిమార్క్స్ ఇవ్వవలసిందిగా కోరుతున్నాను. థ్యాంక్ యూ and all.
Thank you, sir. Thank you, sir, for your welcome address. Once again, warm good afternoon to our Honorable Vice Chancellor, Professor Mokka Jagannath Rao Garu, our respected Registrar, Professor Battu Gangarao Garu, our beloved Principal, University College of Science and Technology, Dr. K. Rameshwari Madam, Principal of University College of Arts and Commerce, Professor K. Ramesh Garu, and other Executive Council members, dear faculty colleagues, and students and participants from various colleges and other organizations. COVID-19, coronavirus disease in 2019 is definitely a nightmare to all of us, that is scaring us from about 5 months. Somewhere I read that the most negative word in the year 2020 is positive. Yes, being Corona positive. We all have locked up ourselves at home from March onwards with the hope that this disease, the spread of this coronavirus may get controlled in next coming few days. All the media has also spread the news that in the coming few days, we are going to arrest the spread of this coronavirus and all our activities are going to be resumed normal. But still, it is uncontrollable and unclear. And every day we are seeing the cases have been multiplying dramatically. This pandemic has severely infected many people across the world. Many patients have lost their lives after suffering in hospitals. And there are many other side effects also of this COVID-19. Several people were stuck jobless away from their native places, and we have already seen the pathetic condition of migrants with our broken hearts. And even students are also clueless about the completion of the academic year, completion of the examinations, announcement of the results, joining in new courses, etc. But there is a saying, the other side of this COVID-19, there is a saying in English, blessing in disguise. So this COVID-19 and the lockdown period has also given us certain opportunities that we could not make them in our daily chaos of moving here and there with our routines of job opportunities and activities. We have seen that in our previous decades, the depletion of the ozone layer, which is causing serious harm to our environment. And within the time of these 3 or 4 months, okay, it is observed that the ozone layer has been rejuvenated a lot because of this drastic cut down of the automobile exhaust and decrease in the pollution because of the arrest of the industrial activities and all. And not only that, this lockdown period of 3 or 4 months has given us a good amount of time to spend with the activities like meditation, yoga, spiritual activities, and also indulging in some other academic activities. And with the encouragement and support of our most dynamic and visionary Vice Chancellor, the faculty members of our university have been continuously arranging certain kind of programs related to academics, are creating awareness among the students and people. We are organizing a series of events, and this event also will come under such a category. When I approach our Vice Chancellor with an idea of conducting a webinar, he encouraged me so much and asked me to go ahead. And for any kind of help, he is always ready to extend. And after getting the idea of organizing a webinar to create awareness and to educate people about this COVID-19, it happened to listen to the lectures of Dr. D. Raghunath Rao Garu as well as Professor Suresh Verma Kuchipudi in some other events. Then immediately I have considered them to be a part of this international webinar. And when I approached them to deliver a talk in this event, they have accepted without any hesitation. So I must be very thankful to Dr. D. Raghunath Rao Garu as well as Professor Suresh Verma Kuchimudi for accepting this invitation and for being part of this webinar and sparing their valuable time despite of their busy schedules. And with this few remarks, now I want to go into the main program. So the program begins with the addressing of our Honorable Vice Chancellor to today's gathering. And before our Vice Chancellor speaks to the web gathering, I would like to say a few words and a brief introduction about our Honorable Vice Chancellor. Sir, if you permit me, I would like to take the opportunity, sir. Professor.
Let us go ahead with the program.
Sir, please just for a few minutes, sir. Sir, Professor Mokka Jagannath Rao Garu is a professor of geology in Andhra University and has now working as the Vice Chancellor of Adhikavi Nanya University. Is a very keen academician, very enthusiastic researcher, and a very good human being. Since he has taken charge as the Vice Chancellor of our university, he is heading us with a vision to see Adhikavi Nanya University at international level and should always be the best choice for students to go for education, research, training, and consultancies. Coming to the brief biodata of our esteemed Vice Chancellor, he got his master's degree in engineering from University of Alaska, USA, and PhD from Andhra University in the year 1985. Despite of getting selected for many national level job opportunities, including UPSC Civil Services, UPSC Geologist Examination, in which he secured All India 11th rank, he has chosen to join as assistant professor in the Department of Geology, Andhra University in 1986, only because of his true passion towards teaching. Later, he got promoted to professor and also the head of the Department of Geology. He is a serious researcher and academician, having more than 33 years of vast experience in teaching and research. He has published nearly 150 papers in national and international journals. 33 scholars have been already awarded PhD under his esteemed guidance. His students are not only from geology, but also from different disciplines like computer science, geoengineering, geophysics, and allied physics. This is a rare thing that we actually observe. And coming to the research collaborations, SAR has very strong research collaborations with various organizations from USA, Canada, and Australia. He has completed 8 major funding projects with the leading funding agencies in India, including DST, UGC, CSIR, DOD, APMDC, ISRO, and also ONGC. He got huge consultancy projects from ONGC, namely studies on Holocene evolution at Mahanadi Delta and studies on land subsidence due to hydrocarbon exploration and exploitation at KGPG base in Andhra Pradesh using remote sensing and field studies. He has held many other administrative positions also. To name few of them are Director of Information Management Center, Andhra University, Dean for PG and Professional Examinations, Andhra University, Director for International Student Affairs, Andhra University, Director for Delta Studies Institute, Andhra University, Chairman for Board of Studies, Department of Geoscience, Ambedkar University, Srikakulam, and also Head of the Department of Geology. He has filed a patent recently also entitled an inventive model that explains the genesis of Bay of Bengal and Arabian Sea, which is under consideration. He got many awards and honors, including Dr. Sarvepalli Radhakrishnan Award, the Best Academician Award of the year 2019 by Andhra University. He was also awarded as a Scientist of the year 2017 by National Environmental Science Academy, New Delhi. He was in the editorial board of many reputed journals, and Indian Journal of Environment and Eco Planning is one of that. He is the member of many professional scientific bodies of national and international repute, and also as an accessor, is a member of various boards of selection committees at national and international levels. So these are the few lines I would like to say about our Honorable Vice Chancellor. And beside all these, I can say one thing that I have observed is, being a Vice Chancellor of one university, that is Adhikavan Nanda University, Sir always feels bad for not spending time with his students and research scholars, because that is the thing he likes mostly, to involve his students and research scholars into guide into this geology and other allied fields. So that thing is very, very rare. And Sir comes under such a category of rare Vice Chancellors. And it is a very great pleasure for all of us to be led by our visionary Vice Chancellor. Sir, now I request you to address the gathering.
Thank you very much, Dr. Deepthi, for your kind words of introduction. And I'm very happy that you expressed your words of affection and liking for my administration. And this is a great opportunity to be part of this one-day international webinar on entitled COVID-19, Contemplations on its Pathophysiology and Possible Ending. And today, I'm extremely happy to see my registrar, Professor Ganga Rao, and EC members, Professor K. Ramesh, Professor Jaganmohan Reddy, Professor Ashok, Principal of College of Science, Professor Ramaneshwari, and the convener of this program, Dr. Deepthi, and other principals, deans, officers, participants, and especially today's keynote speakers, Dr. D. Raghunath Rao, Professor Suresh Verma Kuchipudi, and others. When I see this title of today's webinar, I'm really relieved to know this webinar title is focusing on the most important aspect of the COVID-19, which states its possible ending. We are really excited because every one of us are worried and looking forward when it will end. Since this crisis, the corona crisis came, COVID-19, we are all clueless, especially we are non-medical professionals, we are laymen, but we are looking forward to all the medical professionals, all the scientists, all the pathologists, all the microbiologists, and all other professionals who are really working hard on this pandemic, COVID-19, to make a vaccine and see that the entire globe and the entire world is relieved from this pandemic. For the last several months, we are literally locked down and confined ourselves to our homes with a lot of stress, worry, and there is no, it seems, it is an unending problem. Despite all these things, we being the academicians and being the professors and researchers of Adhikavin Annaya University, we don't want to get depressed. We want to take this academic program forward. I requested all my faculty to take this as an opportunity instead of a problem. So let us use this online and the internet and the virtual reality as the advantage and do the academic activity, academic events in the form of online teachings, online webinars, online faculty development programs, etc. I'm very thankful and I'm very happy to know my faculty that they are very active, they're doing excellent work, and in fact, in every department, at least one or two programs have been conducted, and there are many departments are coming forward to conduct many more such events. So in this way, the COVID-19, the corona crisis, has given us an opportunity to explore this online and internet facility to be used in academic activity. So in future, we can use this platform along with the regular physical teaching in classrooms. What the government is telling, so blended teaching. So teaching in a classroom, teaching in online. So we need to look forward for such platforms in future and make them more effective, more useful for extending the knowledge to the students remotely. When I come to the coronavirus, so when we know, every one of us know about the story of corona very well. There are many apprehensions and many miscommunications about this corona because of the lack of proper information, because we are fighting with an unknown enemy, the virus, which is not visible to the naked eye. Being a layman and we are but educated professionals, we know that it has been, the sources have been the city of Wuhan in China. And initially, we have not taken proper precautions. Had it been confined to Wuhan city itself, there and then itself, we could have contained this corona there only and could have saved the globe from this kind of pandemic. So we are not here to blame anybody, but it is the biggest lesson to the mankind. We should know and we should be ready for this kind of eventuality. This is a lesson for us to be prepared. We should have a preparedness for such pandemics in future also. I wonder why all the organizations related to these kind of pandemics do not have any preparedness for such pandemics. Is there any way we can have a global platform so that they'll be continuously working on such possible pandemics in future so that the suffering can be reduced? Once the pandemic is initiated and spread at the global level, doing something for a vaccine, which is taking 6 months, 1 year, testing is going on. laboratory and clinical tests are going on. These kind of sentences and words are giving some kind of depression and panic among the public. So what I want to say is, but the global community, the scientific community related to these medical professionals and the biochemists and pathologists should have a platform on a continuous basis. They should be working and they should prepare some kind of, they should have a plan for dealing with such pandemics in future. So this should be taken as a lesson for all of us. Initially, they said it is not transmitting from human to human. That is one miscommunication. Later, they said you have to be having a social distance and having a mask, close your mouth and nose, don't touch anybody, keep away. And later they said it may transmit through ice also. So after 2 months, they said like that. And later they said it will be transmitting, it will be transmitted maybe up to 5 feet or 6 feet. When I see in the internet and when I see some papers, I have seen the size of this virus is almost 1/10 of a micron, 0.1 microns size. I wonder as a scientist, is it not airborne? Why it only travels 5 feet? Even a sand grain will be traveling a kilometer and kilometers to kilometers. The sand grain is much larger than this virus. So these things need to be understood, need to be studied by all these experts. And I always say that in future, we should have a contingent plan to tackle such pandemics, to save the humanity. We cannot wait for 6 months or 1 year or 2 years to get a vaccine. At least some kind of relief should be given, or some kind of program should be there to stop its spread or slow down its spread so that we have enough time to protect ourselves and enough opportunity to save lives and save the economy, save the daily life, save our regular lifestyle. Anyway, I hope there are two experts, Dr. D. Raghunath Rao, Professor Suresh Verma Kuchipudi is a clinical professor, head of microbiology. Penn State University must be having more knowledge and technical information on these questions. Whatever it may be, this is an opportunity for all the youngsters to learn from these experts, to get their doubts clarified and know what is happening at global level to develop a vaccine or contain this disease, etc. And especially, we are looking forward to see how fast this will be ending. So the possible ending, that part of the title, I like it most. Thank you very much for giving me this opportunity. Thank you.
Thank you, sir.
Thank
Thank you for addressing the gathering, sir. Yes, really, all of us are eagerly waiting to know when this COVID-19 will come to an end and how it is physiologically interacting with human being, that it is becoming that much deadly. I hope our esteemed speakers will make us clear about all these questions. And now I request one of our speakers, Dr. D. Raghunath Rao Garu, who is the founder director of Homi Baba Cancer Hospital and Research Institute, to deliver his talk. And before going to listen to him, now I request one of my colleague, Dr. C.H. Padma Rao Garu, to introduce Dr. D. Raghunath Rao Garu to the gathering. Padma Rao?
Madam, this is Vidyasagar, madam.
Sir, now Vidyasagar will take the opportunity of introducing Dr. D. Raghunath Rao Garu to the gathering. Sir, please go ahead.
Madam, good evening to all. This is PSR Vidyasagar from Department of Chemistry. It is my great pleasure and honor to introduce today's first speaker, Professor D. Raghunath Rao Garu, to this international webinar. Professor D. Raghunath Rao Garu is presently working as a director, Homi Baba Cancer Hospital and Research Center, Aganampudi, Visakhapatnam. He completed his MBBS degree at Andhra Medical College, Visakhapatnam in 1981. He done his master's degree in general medicine at Punjab University, PJIMER, Chandigarh during 1982 to 85. He done his doctorate of medicine in medical oncology at Cancer Institute, Madras during 1987-89. He worked as a professor of medical oncology at NIMS, Hyderabad during the years 1989 to 2013. So fellowships and awards, he got Lady Tata Senior Research Fellowship and ICMR Senior Research Fellowship during his MD course. He got the fellowship of Royal College of Physicians of London in 2018. He got International Union Against Cancer Fellowship in Bone Marrow Transplantation, Princess Margaret Hospital in Toronto, Canada in 1993, and also International Union Against Cancer Fellowship in Palliative Oncology at University of Birmingham, UK. He was Honorary Senior Research Fellow in Clinical Pharmacology, Oxford, UK, and member of the Indox collaboration during 2005 to 2009. He got Dr. B.C. Rai Award in 2016. He elected as a Fellow of Andhra Pradesh Academy of Sciences 2014, and he is the certified member of European Society for Medical Oncology and also clinical trial investigator of the American Association of Clinical Research Professionals and the Indian Society for Clinical Research. He was the member of the International Advisor Committee of the American Society for Clinical Oncology during 2007 to 2010. He got the preceptorship of the International Chronic Myeloid Leukemia Foundation at the Oregon Health and Sciences University, Portland, USA, and he has 25 years of teaching experience, and he published around 137 papers and 4 textbook chapters to his credit. He acted as an examiner and inspector for DM and DNB, that is Diplomate of National Board Examinations in Medical Oncology since 1996, and he was the editorial board member of in Indian Journal of Medicals and Pediatric Oncology, South Asian Journal of Oncology, Indian Journal of Cancer. He acts as a national coordinator for training module on cancer prevention and screening and non-communicable diseases control program, Government of India, and he is the founder of Medical Oncologists Forum 2008, and his areas of interest are cancer education and training and drug development, translational research, clinical trials in cancer. So this is brief introduction about Dr. Raghunath Rao Garu. So thank you. Thanks one and all. Thanks to Dr. Deepti Madam for giving this opportunity. So over to Deepti Madam.
Thank you, Vidyasagar sir. Sir, Raghunath Rao Garu.
Yeah. Good evening, everyone. First of all, I would like to pay my regards to Professor Jagannath Rao Garu, the Vice Chancellor, and I would like to thank all the senior administrators as well as the faculties, including Dr. Deepti and Dr. Vidyasagar Rao for that introduction and the opportunity to interact with all of you. What I shall do today is to take you through the pathophysiology of COVID-19 infection, and then through potential targets for our treatment program. So today's brief is about this, and I have structured my talk on these lines. A little bit about the structure of the virus, which is important to understand because we need to know how and where we can attack the virus. 2, we need to know what it does to the human body so that we can take care of what we can do to the virus and how does the body react to it. Then we need to know what are the areas in which we can actually launch a pharmaceutical or pharmacological directed attack on it, and what are the possible targets. When and how do we use such targets? So this is in brief today's agenda for this talk. So this is the transmission cycle of the SARS-CoV-2 or coronavirus 2. So it probably originated in bats, from which we got it through the pangolins. So the bat has a very special way of dealing with many viruses, not necessarily the coronavirus. It simply doesn't react to it. So just remember this small point. The bat does not suffer from the virus. So also the pangolin, that is the anteater, which also doesn't suffer from the virus. We'll come back to why they don't suffer from the virus, why the humans actually do very badly. And pangolins were a delicacy, and so man is purely an incidental host, simply because he has this angiotensin converting enzyme 2 receptor on many cells in the body, including the respiratory epithelium. Now, our Vice Chancellor has made a remark that if dust particles can travel several kilometers, why can't, which are larger than this virus, which is about a fraction of a micron, why isn't this actually transmitted so far away as what, just about 6 feet or a meter? The reason is viruses cannot survive outside a host. That means they require a cellular environment to survive. So the longest they can possibly survive is when the surface is wet, humid, and there is no exposure to sunlight. So if you expose it to sunlight, the ultraviolet rays in the sun very actively kill it within a period of about 30 minutes to 2 hours. So even if it were to travel in air far away, it can only travel through a droplet spread. That means whatever happens when we cough, when we talk, when we sneeze, or forcefully exhale, what is so-called aerosolization. So the droplets actually pick up the virus from the nasopharynx, that is from behind the nose, and then take it along with it. So the larger droplets very quickly settle down within a meter or two. That is why we said if the 2 meter distance can be observed, it is more than enough. So it is now going on through human to human transmission. Whether we can transmit it to animals, possibly yes, we seem to be able to transmit it to household pets. So we know that we can demonstrate it in household pets. Do the pets suffer from the virus? We are not yet sure whether they also develop COVID-like symptoms. None of them have been reported, but definitely many household animals as well as farm animals seem to contract it from the human host. So please remember that it is not going to end with human beings alone. Now, we need to understand what is so important about this virus. Some people, you know, you may have read it on the internet or in the newspapers, that there is a large amount of what theory I have given you in the previous slide, that it came from bat to pangolin, pangolin to humans, is shown here to the civet cat. So the other three, the bat, pangolin, and civet cat, are wild animals from whom we have contracted. So you see in this genomic homology map that these are the elements which have come from the human beings, which are common to the human beings, which have come from the bat, which have come from the pangolin, and which have come from the civet cats. So HIV, I mean, just like the HIV virus, which is another RNA virus, or the HCV virus, which is another RNA virus, as they leave the host cell, they pick up bits and pieces of genetic information and take it with them. So these are the elements in its genome which belong to each one of its former hosts. So there are elements which belong to bat, there are elements which belong to the pangolin, and there are elements which belong to civet cats. So we know that the genome has some homology elements with the three animals which I have depicted. So it proves that yes, the transmission probably occurred in the fashion which I have depicted. Coming back to the SARS virus, it's a very simple virus. like most viruses, it has an inner nucleus, which is a bare single-stranded, straight or linear RNA strand, and then it is covered by a membrane. I will come back to the membrane a little later, which we call the and then a thick proteinaceous envelope, over which we have some spikes. So it is the spikes which resemble a crown, and then that is why it is called, these viruses are called coronaviruses. There are many coronaviruses. Most of us are possibly very familiar with mumps. Most of us may have suffered from mumps when we were children, enlargement of the parotid glands, which are painful. So that is also a coronavirus only. Some of you who may be working on the veterinary side may be very familiar with distemper. So which is called canine distemper, which is also an RNA virus. So this is a structure which is very common to all of them. So it is important to understand that we know the structure and it is completely elucidated. There is nothing to worry about whether we know the structure or not. We know the structure from the coronavirus 1, which caused the MARS, that is the Middle East Respiratory Distress Syndrome. So it is very much homologous to that structure. Now, looking at it in a very different fashion, you have an RNA which is sitting on a scaffolding of nucleocapsid. So the small yellow spheres are the RNA virus, and then that is sitting on a capsule, scaffolding called the nucleocapsid protein. And then, as I told you, it has an envelope which I have depicted in red color, and it has several envelope proteins, two of which I have depicted here. One in pink, which is called the envelope protein, and the other is called the membrane protein or the M protein. Most importantly, it is the trimeric spike protein or S protein I have depicted here, which is very important. That means it is like a trident, something like a trishulam. So it is called a trimeric S protein. So it is this S protein which helps it get internalized through an exosome series. So this is the single-stranded RNA genome of the entire SARS-CoV-2 virus. It's about 30 kilobytes length, extraordinarily small, but seems to be extraordinarily potent. So if you take it it's much, much smaller than any known particle which we know. So it has some proteases. It has the necessary element of replicase and helicase, and then it has an endoribonuclease, and then it has something which manufactures the envelope as well as something which manufactures the spike protein. Now, what do we need all this for? It is to make a dent or a hole or maybe a rent, if you want to call it, in the membrane of the host cell, and then fuse with it. The envelope fuses, the envelope doesn't enter the cell, and only the RNA is injected into the cell. This is what I have depicted here. If you take the ACE receptor, angiotensin converting enzyme 2 receptor, there are two receptors. This is the 2 receptor. So it interacts, the spike protein interacts with the ACE2 receptor, and the membrane fuses with our cell membrane, and only the RNA with its nuclear RNA sequence and the nucleoside structure is injected into the cell, and within the cell it uses the cell's assembly structure to translate, and then after the translation it undergoes structural protein synthesis. It uses the entire machinery of the cell, assembly and then budding in the Golgi apparatus, and then it escapes out as through an exocytosis. This is purely an endocytosis. This is an exocytosis. This escapes out as a newly made viral particle outside the cell. So this is the traditional life cycle of the coronavirus within the host cell. Now, interaction of the viral protein with the angiotensin converting enzyme is important to understand. So what is happening here is, this is the host cell membrane, and within the host cell membrane, as I told you, everything else remains outside. Only the nucleic acid is enveloped into with our own cell membrane, and like a vesicle, it is brought into the cell. So it is the human host membrane which actually does this task. So the spike protein, after it induces, it allows only this part, that is the RNA, to enter into the stride. So this is the structural domains of the RNA. So what it means is, it contains elements which are essential for it to bind, essential for it to cause lysis, essential for it to disrupt its own envelope, essential for it to inject it through a process of endocytosis. So all of it is available in that small little particle. Okay. Now, what do we know about this? Anything further? Yes, we know that the entire assembly is happening because it's an RNA virus. It happens only within the cytoplasm of the cell. It never integrates or enters into the nucleus of the cell. So it never interferes with our genome. This is important to understand. So we have a replication transcription complex, and then a genomic and subgenomic RNA synthesis, and then a genomic RNA replication, and then that is led to a translation of viral protein structures. So it needs to synthesize all the elements which form the viral particle, not just replicate the RNA. It needs to synthesize its envelope, it needs to synthesize its nucleocapsid, it needs to synthesize the entire spike proteins and the membrane proteins. All of it is done by the cell, and then it escapes out of the cell, ready to infect another cell in the body. So this is important to understand. So let us see what is the clinical presentation of patients with COVID-19. So I have given you here some which are common to all the viral infections, including the influenza virus infection. On the right side are those which most of us will present. On your left side are those which are nearly fatal. So a fever, a headache, a cough, a myalgia, that is body pains and muscular pains, are common to any viral infection. Hemoptysis, that is bringing out blood in the sputum, breathlessness, pneumonia, septic shock, renal failure, diarrhea, and a coagulopathy, which is not depicted in this slide, are all features which are very specific to the respiratory viruses. That's the reason why we called it COVID-19, since we had several virus infections in the past, including the Middle East respiratory virus, the Ebola virus, etc., which also caused similar syndromes, which is exactly the same as the others. So this is a part of our reaction to the infecting organism. So we can divide this into three types of spectra. One, which is very mild disease. I have underlined what is important is 81% of the cases show very mild disease. The second is severe disease. Don't worry about what all I have written there. 14% of the patients will show a severe disease. Critical disease, this is beyond the severe disease, is found in about 5% patients, and these patients have respiratory failure, septic shock, multi-organ dysfunction or failure, and about out of these 5%, the total number of patients, 3% die. So that is the mortality figure for the coronavirus, which is far, far lower than the seasonal influenza virus infection, which we normally encounter. So although most of us this virus, the reason why we are not too worried about this virus, although it's highly infectious, but it is not so fatal as majority of the common seasonal influenza viruses, which are adenoviruses. So we classify COVID-19 patients into asymptomatic COVID nucleic acid test positive, which most of us see without any clinical symptoms and signs on the chest image, and the chest imaging is normal. These are called asymptomatic patients. It can be a rapid test or it can be a RT-PCR, whichever is positive is fine. Then some have mild symptoms, as I said, fever, fatigue, myalgia and cough. Some have sore throat, runny nose, sneezing. Some also have symptoms like the original influenza virus, like nausea, vomiting, tummy ache, what we call intestinal flu. So it can also occur. Moderate is pneumonia with a cough, but there is no lowering of the oxygen carrying capacity, and CT scan may show some chest lesions. Once the oxygen carrying capacity drops below 92%, most of them are critically ill. And of course, those who need a ventilation are the severe and the critical patients. The critical patients have hypovolemic shock, encephalopathy, a heart failure, and a coagulation dysfunction. This is something which is strange, which we did not observe in other COVID infections, and a renal injury. That means a multi-organ failure seems to characterize this group of patients. So this is it doesn't affect the type 1 nematocyte. It affects the type 2 or the alveolar cell type 2. This is actually a human cell which is isolated and cultured in vivo and then infected with the SARS-CoV, and the viral particles you can see as double membrane particles. So inside and outside, they're leaving outside the cell. So all these particles are, this is inside the cell, these particles are inside the cell, these particles are outside the cell. So vesicles in the type 2 and along the apical microvilli along the nematocyte. So let us see what does it do. What is the pathophysiology in the lung parenchyma? This is important to understand. So as I said, it infects the airway cells, it infects the nasopharynx also, but that doesn't do any damage. Then on one side, on the upper side is the oxygen exchange site, on the lower side is the capillary endothelial cell. So here is the macrophage. Now the trouble starts when the infected cell is recognized by the alveolar macrophage, or what we call antigen processing and presenting cell. So this is what is the infected cell. Virus replication, then pyroptosis. Pyroptosis is the cell just drops dead. There is no cell after the virus particle leaves. It's an empty shell which we find there, a blunt shell. So the greater the amount of injury to this lung, the greater the number of dead cells in the lung parenchyma. This is important to understand. Now, the tragedy occurs when this macrophage starts asking for help. So it releases several chemical messengers into the blood, asking all these cells to be recruited. So it could be interleukins, or it could be macrophage activating cells, or macrophage chemotactic cells. So it attracts the T cells, it attracts the monocytes, it attracts the macrophages. Okay, in turn, they return a positive feedback by giving interferon alpha. Unfortunately, I will come back to the interferon. The interferon alpha is the one which actually should regulate. It should prevent the neighboring cells from getting infected with the virus, but that doesn't seem to be happening. So we seem to have, the virus somehow seems to have blunted our ability to release interferon alpha and gamma into the cell, into the blood. So now we have two varieties of reaction. One which is given here in green is called the healthy immune response. The healthy immune response probably occurs in those who are asymptomatic are mildly symptomatic patients, not in those who have severe symptoms or critically ill patients. That is what happens in dysfunctional immune response. In the dysfunctional immune response, we have the host reacting in an overwhelming way. That means the immune reaction from our body overwhelms the entire system, in that it starts destroying everything in its sides. So it attacks not just the lung, the normal cells in the lung, it attacks the capillaries, it attacks the blood vessels, it attacks the brain, it attacks the heart muscle, it attacks the kidney, it attacks the liver. So all the organs, as if something has gone haywire, it is like spinning something like a artillery armor firing all around you. That means you kill yourself in your attempt to kill the enemy. This is exactly what happens when the dysfunctional immune system, with excessive infiltration by monocytes, macrophages and T cells, systemic cytokine storm occurs, pulmonary edema and pneumonia results, and there is widespread inflammation and multi-organ damage. So we have two responses, either recovery or death. So this is important to understand which one will lead to which. Now, a little bit about the thrombosis and coagulopathy which seems to occur in this condition. This has been recognized just a few weeks back, that there is massive intrapulmonary hemophagocytosis. That means cells are eaten up by our own macrophages, our own blood cells, our own RBC are consumed as if they have been infected with the virus, are consumed and eaten up by our own neutrophils, monocytes and macrophages, and that leads to a lot of, that means it leads any nucleated and non-nucleated cell. So it eats up WBC, it eats up RBC, it eats up platelets. So suddenly a lot of cytokines are released into the cell, and then we have a huge amount of thrombosis occur in small capillaries. I have depicted here in this. Here the thrombosis actually leads to the capillaries being blocked, and since the pressure from the heart is so high, the capillaries break open and there is hemorrhage or blood leaking into the lung. So this is exactly what happens in the lung. In a summary, SARS-CoV enters into the body and then replicates initially in the nasal epithelium behind your nose, then finally enters in the lung and then starts multiplying there. There you can have either antigen presenting cell producing a humoral immunity or a cellular immunity. So we don't seem to be able to demonstrate this. So we are unable to demonstrate this. A few minutes back, there was a paper which said that reinfections are possible because we seem to be mounting an ineffective immune response, and that ineffective immune response seems to last just about 8 weeks to roughly about 24 weeks. So the maximum time it has been documented has just been 24 weeks. Beyond 24 weeks, it looks like we are likely to lose the immunity developed in our body. So what are the therapeutic approaches against the SARS-CoV? I will talk a little bit about it. So we can mount antibodies to the spike, which may be from a vaccination or an adaptive transfer which can block the binding of the virus to the ACE2 receptor. We can, of course, like we use for human immunodeficiency virus or to the hepatitis C virus, several proteases which are repurposing. Remedies, you might have heard about it, which is now being used as a protease inhibitor. It blocks and prevents the enzyme from activating the spike protein through a protease cleavage. Or we can use a virus-specific effector CD8 cell arising from a vaccination with a suitable antibody directed against it. So it requires a combination of humoral and cell-mediated immunity to effect an antibody-directed cell-mediated cellular immunity. Or possibly we can remove it by mechanical filtration all the pro-inflammatory cytokines. The fifth one to join this after I finish the slides is low-dose radiation therapy. Just about 40 centigrade, which is about 1/4 the dose which you might receive when you have a CT scan done to your body. So that amount of radiation to the lung seems to allow patients to recover. The All India Institute of Medical Sciences in New Delhi has already started this as an experimental therapy, and the results are very gratifying. Severely ill patients who were given radiation to the lung to stop all the inflammatory reaction from occurring seems to recover very quickly. So, treatment options left to us are drugs which seek to neutralize the virus itself, which can alleviate its life-threatening symptoms and to boost our immune system to fight off the infection. So, the treatment goals will be to treat the symptoms which occur, treat the inflammation which occurs, retard or completely stop the antiviral growth, and of course, as our Vice Chancellor said, prevent it from infecting anybody. So, the squares actually represent how effective or ineffective we are. At the moment, we are able to prevent it only in about 1/10 of the way by purely social distancing, hand washing, and using a mask. That means we are not giving the virus a chance to infect another host, but we are not at anywhere near developing a vaccine. So we have several clinical trials, more than 30 odd innovations are yet to go into clinical trials, but none of them have actually entered into a standard practice. All of them are purely in the emergency use situation only. So these are some of the treatments which are available. Don't worry about what they are. All of them are directed towards each one of those cytokines, each one of those enzymes, which we know are useful for a RNA virus and which can actually be used. So I have listed some 40 odd such drugs which can be used. Some of them are purely hypothetical. They are not yet even in the preclinical stage. I will come back to some basic vaccine design. All vaccines must contain two components: the adjuvant, a molecule that acts as the danger signal to activate your immune system. That means something needs to kick the immune system to say the payload I am carrying is dangerous. And then the antigen itself, which is the unique molecule that acts as the target for the immune response to the virus. The most unfortunate thing is so far is we have been unable to identify what is the best antigen. That means what is the best target for us to raise an immune response against. So I will show you what are all the methods in which we can do it. But we must understand this must result in an immune response which produces antibodies, which is humoral immunity, which bind to the surface of the virus to prevent the infection of the cells. Something like what we do in poliovirus, or generate T cells which kill the virus infected cell, not the virus, it kills the virus infected cell. We are already doing it, but the body is already overreacting to the virus. So we must be careful whether we will be able to achieve a via media between a hyperimmune response and a normal immune response. So first we need to select a target or an antigen on the virus and an adjutant which makes the body recognize that this antigen is what produce the vaccine, and then you can have two responses, a T cell response or a B cell response, which is an antibody or humoral response. Here the antibodies block the virus from entering the cell, and here the T cell mediated immune response kills the infected cell. So let us see whether we can raise an antibody against one of these. Yes, we seem to be able to use any one of these which I have mentioned as an antigen. So we can use the spike protein, we can use the membrane protein, we can use the capsid protein, we can use the viral RNA itself as one of the antigens for our vaccine. And we can also block perhaps the angiotensin converting enzyme 2 receptor using drugs or an antibody or vaccine against it. So, as I said, the virus candidates can be the virus itself, the nucleic acid of the virus, a viral vector. That means you use another, you introduce it into a vector and multiply it and raise an antibody, and then use that to block. And then, of course, we can have a protein-based antibody that is directed against the protein of the virus. So, all that can be used for as an antigen for raising an vaccine against the coronavirus. So, this is a basic diagram. So, once again, recapitulate the same. So, you need to have the something on the coronavirus for you to use, and then you can stop it at any level you want, from entering, from replicating, from getting translated, and then integrated, and then or preventing the body itself from overreacting to the virus itself. Okay, so this is how each one of them acts. That is how the basic. This is how the nucleic acid bound vaccines raised against nucleic acids can work. So it can work both for a DNA virus or you can use it for an RNA vaccine. So this is important to understand, so that we can raise it against the lipid coat against in which they are encapsulated and brought into the cell. So this is important that we can do it. Viral vector vaccines, you have a replicating viral vector, something like a weakened measles virus we can use, or you can use an endovirus like an influenza virus, which is a non-replicating viral vector, and you use them together because the body knows what it is. So it raises an immune response against that against the virus, against the spike gene, or against the inactive element, and then we can synthesize it. We can also use protein-based vaccines like the protein subunits. So many teams seem to be working on the protein-based vaccines, and we can also work on empty shells. So without the nucleic acid or the nucleocapsid inside, they are called virus-like particles, which are empty shells which mimic the coronavirus structure, and we can use them. We have successfully used in the human papilloma virus-like particles to immunize girls and boys against getting a papilloma virus. So, there are a good number of vaccines in development. Almost at this moment, about 32 different vaccines are in development. Each one uses a different platform. Some are using the virus, some are using viral vectors, some are using nucleic acid. As I said, many are using protein-based antigens to develop a vaccine. What we need, what is called, I'll tell you a few of them. One is called a quick vaccine. It's a lipid encapsulated mRNA vaccine. It's a short piece of genetic material from the virus, which is in an oily lipid layer. And the lipid helps the mRNA get inside the person's muscle cells, and it provides a blueprint to make the spike protein of the antigen. mRNA itself acts as the adjuvant. So that's the danger signal for the body to raise the vaccine. We can also have subunit vaccine, as I said. The spike protein is used as the antigen mixed with the adjuvant to activate the immune system. The shape of the spike protein must be highly consistent. If it undergoes a drift or a shift, of course, we can't do anything about it. So if it changes, we can't do much about the spike protein and being used as an antigen. Then there is something called molecular clamps, which are short pieces of protein, which hold a larger protein in the correct shape to stabilize the viral protein. Most of you know that the immunological fit is a stoichiometric fit. That means the shape and the size and the dimension and the direction, all of them are equivalent. That means it is important in not just X and Y axis, but it should be equivalent in X and Y axis and Z axis, and it should be either a levo or a dextro form. So you hold it in correct shape, and we have been, these are the successful vaccines which we have developed using a molecular clamp. These are very rapidly done. You can develop it in a couple of months. So we have raised it in respiratory syncytial virus, in influenza virus, in Ebola, in the Middle East respiratory syndrome, which was again a coronavirus, the Lassa syndrome, and the Nipah virus, which occurred in Kerala. So all these use the same molecular clamp to stabilize the protein against which we can raise the vaccine. Viral vectors, it takes genetic material from the SARS-CoV, insert it into a harmless virus, and you give it to a person. The docile viral vector can't cause any disease, but it looks like a vicious virus to the immune system. It generates a robust immune response. We use this also for development of the Ebola virus. Something which was exciting when the whole pandemic started was BCG, and people thought that this actually explains why we did not find so many symptomatic patients in India. Probably the tuberculin vaccine was actually used as a non-specific immune mediation. So it pre-activates innate cells and engulfs and kills the virus, the monocyte cell. So it's important to understand sometimes a BCG vaccination seems to protect many people against the bug. So there are actually trials now being undertaken in Australia and in Netherlands for healthcare workers. They are vaccinating the sanitary workers, the staff nurses, the technicians manning the intensive care units, as well as doctors attending on those patients with BCG to see if it has a very good effect in those people with virus. So not just in our country, many other countries also going. So these are several. At the last count, we had about 61 in the early development phase for candidates, which are vaccine-based, so which are antigen-based and vaccine. We have just completed some phase 2 trials. There are some phase 3 trials ongoing. You can't get it as quickly as you want to fight the pandemic, because it's a matter of ethical principles that it should undergo a clinical trial to prove that it does. So we know that clinical testing can be done in 6 to 9 months, and the clinical phases themselves require another 6 to 9 months to complete. So it is important to understand that you cannot immediately manufacture a vaccine and get children back to school, as many parents are fighting in the Supreme Court. That is not possible. In fact, it will be impossible to do that. So best is to stay home till the virus dies a natural death. If it doesn't have a host, the virus dies. So if the person dies, the virus dies. If it doesn't have a natural, a host cell, it will die. So it is important to understand that if you prevent the virus from getting access to human beings, it will die a natural death. Of course, it may take a long time, but it cannot have a virulence beyond. So we expect that the peak will occur in a couple of weeks, if not in a couple of months, and beyond that, it won't find enough human beings to attack. This is what the epidemiologists have been telling us. So, in summary, this is the treatment paradigm I have given here. The potential therapies here, the clinical symptoms and the clinical signs. So, in the first 2 stages, the viral response phase, we call it, and that it must end, and then the host inflammatory response phase should not be overwhelming. If we go to the hyper-inflammatory phase, we are in serious trouble. So we may reach a point of no return when we reach the hyperinflammation phase. So it is important that majority of us will probably be in stage 1. If we test everybody in this country, we will probably find that we have 1 billion positive patients or positive persons, not necessarily patients. So all of us may have gone through an asymptomatic infection phase. If we have prevented the lockdown, the lockdown probably will bring it down to probably 1 million. So it is a reduction by 1 in 100%. Okay, so we can use some repurposed drugs, some antibodies, some vaccines. We are using them in the clinic. So that is what we have been doing. Still, we find absolute remedies which work against it. We know that if it is a repurposed drug, we are using repurposed drug like hydroxychloroquine. All the drugs which we used against human immunodeficiency virus as well as hepatitis C, we are using in malaria, Ebola, all of them we are using in this viral infection as well. We are not yet there in antibodies. So what we are doing is we are just taking convalescent plasma and giving it to people who are very sick. We don't know whether it works or not. We don't know whether that person who has recovered has mounted any immune response. We don't know how to actually verify that there is an antibody. Antibody against what? We don't know what that antibody is against, if there is an antibody. And of course, we are speculating that we will soon be able to produce a vaccine. So thank you very much for your patient hearing. So if there are any questions which you would like to ask me, or possibly you can text me if you don't want to ask me straight away. You can send it via the chat and I'll be happy to answer them.
Thank you very much, sir. Sir, can you hear me, sir? Am I audible to you?
Yeah, very much.
Sir, thank you for your elaborated presentation, sir, regarding this COVID-19 and its pathophysiology. Now I request the participants, if any one of you have any questions, you can raise your hand and you will be given option.
I have a question, madam.
Yes, sir, please.
Sir, Raghunath Rao, sir. Hello, sir. Sir,
yes, sir.
Yeah.
I can hear you.
Yeah. Sir, regarding this vaccine, I have a small doubt.
Yeah.
So whenever this, suppose if a vaccine comes at 6 months or after 1 year, if possible, so it works for all kind of age group, right from the beginning, 6 months to some 80 years, up to 80 years, it works, sir?
Sir, there is no reason to believe that the vaccine will not work in people who have an intact immune system. So any person with an intact immune system should be able to raise the necessary antibody or humoral immunity or a cell-mediated immunity, depending upon what is the type of vaccine we have used. So if you are an immunocompromised individual, then of course there seems to be a different problem. For example, we know although the infectivity for cancer patients, infectivity for patients with chronic lung disease, the infectivity for other forms of chronic illnesses including diabetes and the elderly seems to be the same, but the mortality seems to be higher. That means we seem to suffer more from the disease and its symptomatology if we have any of those comorbid conditions. And that's the reason why we have been asking them not to go out. There is a question, is it proven beyond doubt that COVID-19 stems from the bats? Professor Teki from Nannaya University is asking that. We can trace coronaviruses to bats. That means if we take a bat and look for viruses, it is very strange that the bat never raises an immune response to a host of viruses in it, including the rabies virus. The bat doesn't die from the rabies virus. We have known that bats harbor rabies virus for a very long time. We know that bats which were captured in Arunachal Pradesh harbor the virus. It is an absolute fact. It is a published fact. So there is nothing to not to suspect that the virus originated from the bat. I have shown you in my second slide that what are the elements in the virus RNA or the viral proteins which point out to the civet cat, to the pangolin, to the bat, and some human elements. So there seems to be some homology available in the virus. I have already told you that RNA viruses are adapted collecting material, nuclear material or RNA material along with them as they leave the host cell. So they don't know when it comes in handy, so they do it. People wanted me to share the PowerPoint. It is up to Dr. Deepthi. I have already shared the PowerPoint with the organizers. I have no copyright over it, so you are free to use it for any purpose you want. I have given the credits beneath each one of the slides. These are slides which I have, as I prepared the talk, I have collected from across the world literature.
Good evening, sir.
Yeah.
Sir, my name is Dr. Uday, sir. I'm working as assistant professor in Vikram Central University.
Yes, sir.
Sir, very nice presentation, sir. It has given overview about the pathophysiology of COVID-19, sir. Excellent presentation, sir. I have a couple of questions, sir.
Yeah.
So first one is this spike protein is a glycoprotein, sir. So what is the possibility of succeeding in making vaccine against this glycoprotein, sir?
Glycoproteins are extraordinarily good for making vaccines against. So there is nothing odd about using a glycoprotein as an antigen. The only thing is the virus should not change the shape or the structure of the glycoprotein. So glycoproteins keep changing. So that means today it wears a gold crown, tomorrow it wears a silver crown, day after tomorrow it wears a diamond crown, and then a combination of these. So if just you have 1 into 2 into 3 into 4, you know the number of permutation and combinations which can occur on the spike protein. So we have 2 different varieties by which a virus typically changes its character. One is called a drift, the other is called a shift. Professor Kuchipudi will explain that in his talk, so I don't want to impinge on that. So far we have not seen a shift. Shift is something which typically occurs with the influenza virus once every 2 years or once every year. A drift is of course what we know about. We have already seen a drift. In India itself, early on it was reported that we had 7 different subspecies or what we call drifts in the virus. So that is all right. So a drift is not a very unduly worrisome fact, but a shift is a very unduly worrisome fact. Dr. Suresh will explain that to you in detail.
One more quick question, sir.
Yeah.
Is there any real phenomena of herd immunity, sir? Or is it without a vaccine, is it possible to have herd immunity, sir?
There is no evidence to prove that there is herd immunity. So it's a well-known fact. No epidemiologist in the world is actually thinking in terms of herd immunity at all. No, it is not possible to develop a herd immunity at all. It is simply you need to extinguish the virus by not giving it a chance to infect you. But what is worrisome against the herd immunity is the fact that there seems to be a reinfection. Again, Professor Kuchipudi will explain this to you. There are a few instances in China where it looks like there was a reinfection possible. So if we take November 1st as the occurrence, so it's already a little beyond 6 months, so 7 months in fact, or 8 months, and we can see a reinfection occurring in a few people. So that means we are not able to actually mount a robust immunity against the virus. So at the moment, we are not thinking in lines of herd immunity at all.
Thank you, sir.
Is there a strong relationship between weather conditions and viral spread? Why are some cities or areas have high mortality? In fact, the mortality is not very high in India. It is just the number of cases. Unfortunately, what you call a case is often very asymptomatic person. So the greater the availability of a specific test, greater availability of laboratory, greater number of people you will find, positive for the test. So I don't know whether you would like to call that a case and isolate them or allow them to remain in quarantine, either home or a hospital or away in a sports stadium. It's up to you. But I don't know that is called a case. The epidemiologist and the microbiologist possibly would like to call that a case, but from a clinical standpoint, it is not a case. So anybody who is asymptomatic can be testing positive. So is that a case? We don't know at all. Why some cities and areas have a high incidence? Well, they have a high rate of testing, they have a high rate of crowding, they have not, they are unable to comply with the so-called home quarantine or home isolation or lockdown procedures, including hand washing and putting on a mask. So if it is crowded, let us say a house has 10 people, we saw several weeks back, several days back, the whole family getting in, testing positive, but not one single of them had clinical infection. That means they never needed hospitalization. Although all the members were actually quarantined in a hospital setting, all of them returned home fully all right. That means they tested negative later. So it's important to understand that testing positive is not a death warrant. So don't take it in that fashion. Some people have asked any home remedy. Well, there are several, Ayush has given you several measures to boost your innate immunity. We know very well that it boosts your immunity. Some of the measures which they have told you are time-tested, time-honored measures to boost immunity. I'm also a reviewer for the International Journal of Ethnopharmacology. I've already reviewed 3 papers for publication on co-administration of Chinese herbal drugs, co-administration of drugs from the Sindh province, co-administration of Unani medicine in Egypt. So these 3 papers have demonstrated very clearly that the recovery rates, the multi-system organ failures were much lower in those patients. Well, the paper from Hong Kong, from China, also investigated and found what are the immune-mediated hyperactivity like, and they have, to their surprise, they have discovered that the so-called storm which occurs in the body is dramatically reduced by the addition of these drugs. So it is not as if it is only Ayush which is promoting it. All over the world, all ethnic cultures which have some folk remedies available to them, labeled as immune boosters, have tested and found them to be very active. All these have been, all the 3 papers have been accepted for publication. Possibly within this week, they will be published. They are double-blind randomized studies with laboratory backup. Is there a vaccine to come up for COVID-19 in a short period? A short period is an untested period. It is not right on our part to expect a vaccine to come out so quickly. Well, you can always volunteer to take a vaccine. So if the Indian industry and the ICMR are going to launch a vaccine, possibly you can volunteer to, I think this is Vijay Sekaram who has asked this question, Mr. M. Vijay Sekaram. You can possibly volunteer to be one of the first persons to use the vaccine. Why can't scientists target human ribosome as a drug target to prevent the virus RNA enter into the ribosomal binding site? Yes, of course, they are not very robust elements. They are not very robust drugs. That means we need to use an antisense mRNA. Yes, there are drugs which we know are effective against ribosomal RNA, which we use in cancer, which we use in precancerous conditions. And we know that they are highly effective. Antisense mRNA drugs are very highly effective. So possibly you are right. You can actually lead with an idea. You may not be wanting to prepare it, but you can give an idea to the world, and the world will test your idea. So please write up all about it, send it to the Warriors Against Corona. There's a group that is called Crowdsourcing Against Corona and COVID, and you can volunteer. For example, I volunteer in translating several informed consent documents into Telugu, into Hindi, into Tamil, into Kannada, into Malayalam, Indian languages. So these are every study needs an informed consent, whatever new drug, new treatment protocol you are doing. So I volunteer in that. Or you can report about many aspects. There are roughly about 185 tasks ranging from basic research to translation to clinical research on social or psychosocial medicine and epidemiology. So you can actually volunteer to be one of the crowdsourcing key persons for the studies in COVID virus. Why doesn't government concentrate on this seriously? Well, you need some scientists to develop this program. So if you know somebody, you can actually do it. This is from Geetham University. Yes, of course you can do it. You can do it. Vaccine like Red Bunny in USA is not working. You mean to say Remdesivir? Remdesivir at least went through a clinical trial. It did not produce an overall survival benefit, but it shortened the time for people who were in both the intensive care unit without having to go on a ventilator, that is non-ventilatory support or non-invasive ventilation, and also those patients who needed to be on a ventilator. That means, and that is the basis for its approval for use in COVID-19. It did not actually increase the numbers of patients coming out alive, but it shortened the duration by 4 days, which is very important. So if you spend 4 days less in the intensive care unit, you are likely to survive a higher chance of survival, probably a 40% higher chance of survival. Dr. Bangar Naidu, yeah, okay. What about mutations? I will leave it to Professor Suresh to elaborate on that. He will talk to you about antigenic drift and shift. So please bear with me. The next talk is his, so he will give you all the information.
Dear participants, if you have any more questions, you can drop it in the chat box, or else you can mail them to Dr. Raghunath.
Yes, we can answer you individually. There is no problem.
So that
and I don't want to delay Dr. Suresh talk. Most of you, I don't know how many of you have heard him before. It will be astonishingly refreshing to hear him. So please bear with me. You are in for a lovely talk on the virus. And thank you very much, Dr. Deepthi, and thank you everyone who has participated. I'm astonished there are 108 participants in the group. Wonderful. I will mute myself now. Thank you.
Thank you very much, sir. Thank you. Sir, I welcome Professor Suresh Verma garu Kuchipudi from Pennsylvania State University, USA. Sir, can you hear me, sir? Okay. So before we listen to him, I request one of my colleague, Dr. C.H. Padmarao, to introduce Dr. Suresh Verma Kuchipudi to the gathering. Padmarao, please go ahead.
Thank you, madam. Suresh Kuchipudi is a professor of veterinary virology and serves as the head of the microbiology lab of the Animal Diagnostic Laboratory at the Pennsylvania State University. He obtained bachelor degree in the veterinary medicine and master's degree in veterinary microbiology in India. He was awarded PhD from the University of Glasgow, UK, for his work studies on the cellular and molecular mechanism of innate host susceptibility and resistance to influenza A virus in chicken and ducks. He has many years of experience in clinical teaching and research. He is a certified specialist in virology and immunology in American College of Veterinary Microbiologist Board. The aim of his research is to generate knowledge that can underpin the development of novel diagnostic tools and intervention therapies for endemic and emerging viral diseases of animals and humans. Right now, his major area of research interest is to study the virus-host interactions with special focus on emerging zoonotic virus such as influenza A, coronavirus, flavivirus, and herpes virus. And the other current major interest is to understand the molecular basis of innate host resistance and susceptibility to avian influenza A virus. He is an adjunct professor in the Tamil Nadu Veterinary and Animal Science University, Chennai, Tamil Nadu, India. To his credit, he has published 57 research papers in various journals. At the present, he is a member in different professional bodies like American College of Veterinary Microbiologists, American Association of Veterinary Laboratory Diagnostician, American Society of Microbiology. Thank you, sir. Welcome you, sir. Thank you, madam, for giving this opportunity.
Sir, Professor Suresh Verma, sir, can you hear me?
Yeah, I can.
Sir.
So can I start now?
Yes, sir, please. Please go
Okay.
ahead.
So good evening to all the participants. First of all, I would like to thank you, Padmarav, for the lovely introduction. And I would like to thank Adikavi Nannaya University for this opportunity, and also congratulate them for this initiative to educate the university and other members with regards to this pandemic, which is an incredible move. And particularly, I would like to thank the Vice Chancellor, Professor Jagannatha Rao, and Dr. Deepthi for coordinating and inviting me for this. So I fully appreciate the opportunity. And I also recognize that after the outstanding talk by Dr. D. Raghunatha Rao, there is not much left for me to discuss. So what I try to do is I try to highlight some of the aspects that are come up as questions. And I also wanted to mention before I begin the talk that this is one of those defining moments in our history that the entire world is facing a pandemic. And naturally so, everybody is talking about this. And consequently, one of the challenges is that then you will see that there will be a lot of virologists that just will emerge in this period. So, and everybody will claim that they know the stuff, and they talk about these things. And the challenge is with this information overload, even people like us who are in academia, we have a lot of confusion about which information is credible and what we need to really believe or what we cannot believe. So the focus of my talk is also considering that this is a really vast topic and I would not be able to cover everything in this 45 minutes to 1 hour. I'll try to summarize some of the key aspects and hopefully answer some questions. So with that, let me share my screen. Let me also check. Can you all hear me all right? Am I audible?
Yes, sir. Perfect, sir.
Okay. And can I also request everybody to mute your microphone so that there is no feedback or noise as we talk. Okay. So I just wanted to start with these pictures that perhaps, although look in black and white, or might be very familiar to what you see in the streets now. Perhaps people are wearing different clothes, and the context is very similar. So the other day I was just walking outside and I saw a bunch of people here in the US playing baseball, wearing masks. And then there are a bunch of youth, as you can see in the bottom picture, wearing masks, and the couple walking in the streets of London wearing masks. So this looks very familiar to us now. But if I gave this talk 4 or 5 months ago, people would have just imagined that this is like a Hollywood mystery or a horror story. And as you, some of you may suspect, these pictures are from 1918, when the world was facing the influenza pandemic, or the so-called Spanish flu pandemic. So after many years, over 100 years, we are here again, a similar but a different virus. It's a respiratory virus that spreads from people to people, and people know back in the 1918 that they need to wear masks to stay alive. And according to conservative estimates, this pandemic perhaps has killed close to 100 million people across the world. So that is the nature of these pandemics, and hence we all have to be very, very careful and take these things seriously. And as I just mentioned, there are several pandemics in the history. So on this timeline, maybe the font is very small here, but I hope you can see these objects. So these are the different pandemics that were recorded in the history, and the size of the object represents what is the estimated amount of human deaths. The bigger the object, the more number of people died. So the green object here that stands out is the Spanish flu, which I just mentioned. This happened right after the First World War, and some believe that, in fact, the World War was ended because of the pandemic, because people were sick and tired and dying, and they couldn't really fight. And the red object here, or the orangey object here, is the HIV/AIDS pandemic, which protracted for a long period of time, still continues. And at the time of this cartoon was made, this pandemic is believed to have killed at least 35 million people across the world. And there are several smaller objects, and in the timeline you see the tiny little object here is the COVID-19, which is still spreading. So pandemics have happened in the past and will continue to happen in the future. And some of these historic human pandemics, the bubonic plague in as early as in 13th century, which again is believed to have killed 200 million people across the world. So there are other things like smallpox, Spanish flu, and so on. And when we moved on, and we have learned lessons, and we still continue to learn lessons from these past pandemics and the one that is currently rocking the world. So just a little bit of background is my lab studies these viruses, and I've been in this field for over 20 years now. And we study especially a group of viruses that are called as zoonotic viruses. Those viruses that transmit from animals to humans. And the list includes a whole host of virus families, such as influenza virus, coronavirus, and flaviviruses, so things like Zika, dengue, and West Nile, and so on. And we study these viruses in a very specialized lab that are not normally found in many places, and these are called as biosafety level 3 lab. And often friends of mine say, when I say I'm working with the COVID virus in the lab, they'll tell me, oh, you got to be careful because the virus is dangerous. Actually, when we work with this virus in the lab, we actually feel more safe than being to a grocery store or a shop, simply because this lab is a very highly equipped lab with pretty much negative pressure inside. And the people who work in the lab are wearing like the space suits. So although we literally are holding the live SARS coronavirus in our hands, but we feel very safe because the personal protective equipment is so complex that you know for sure that there is no possibility of getting exposed to the virus. So now, again, this might be a repetition to most of you, and I keep mentioning these things because there might be people that are not fully clear about these terms. So I always want to introduce these terms so we are all clear and on the same page. And the terms nCoV 2019, COVID-19, and SARS-CoV-2 are interchangeably used. So quickly, when this virus emerged in 2019 in China, based on the genetic sequencing, it was quickly recognized that this is a completely new coronavirus that we did not know before. And hence it was called as novel coronavirus 2019 because it was found in 19. And subsequently, there are two international bodies that you need to know. The first one is the World Health Organization. It developed a system called International Classification of Diseases. So this system will name the disease so that the entire world is calling a disease with only one name. And before this system existed, if you have studied infectious diseases, we always teach students that this disease was also called by 10 different names or 15 different names. And this is simply because back in the day, in each region, in each country, people gave the name the disease. Even in Andhra Pradesh, we know several pox viruses or pox diseases are often attributed to a certain goddess or something like that. And to avoid such confusion, now the system exists. The International Classification of Disease has officially named the disease caused by this virus as the coronavirus disease 19, in short, COVID-19. And there is another international agency called International Committee on Taxonomy of Viruses. So this agency comprises of experts from across the world, and they study the viruses, and based on the characteristics of virus, they will classify them in a certain family or a genus, and they will also officially give the virus a particular name. So the ICTV has named this virus, which is causing COVID-19, as the severe acute respiratory syndrome coronavirus 2. And the reason it is called 2 is some of you know that there was a SARS virus in 2002 in China. And this virus is very similar or very closely associated with this virus in terms of its genetic composition. And hence it was called as severe acute respiratory syndrome coronavirus 2. So keep in mind, if somebody mentions COVID-19, you are not talking about the virus, but you are talking about the disease. And if somebody mentions SARS coronavirus 2, the mention is to the virus. And this is really important because, especially in the scientific world, when you talk about these things, we need to make sure what we are referring to. So the virus and the cause of the disease. And this is not new. So for example, you know, HIV is the virus and the AIDS is the disease. So there are different names for the disease and the virus, and this is not uncommon in the field. Again, this is a question that gets asked, especially in the US. So I keep always showing this particular slide so that people really understand. And I have been studying influenza viruses for a long time, or flu viruses. And some people will be, may not be now, but in the early part of this pandemic were very dismissive and basically say, oh, this is just like a flu virus and you're just making a big deal out of it. And we need to get on with our lives. So the data speaks rather differently. If you look at the graph on the left-hand side, this is the death percent of people died due to a flu virus based on the age groups. So in the highest mortality rate is less than 1%, and that is in people who are 65 years and older with the influenza virus, also called as seasonal flu occasionally. And this graph is the COVID-19, and the data is still emerging, but still based on when this article was written, you can see that in any given age group, the mortality rate, meaning the proportion of people who would die from the number of people who are infected, is significantly higher in any age group when you compare with this. So the highest can go up to 14%. So clearly, and the reason for this is that the entire world is immunologically susceptible. As Dr. Raghunath Rao has mentioned, none of us have antibodies to this virus because our bodies have never seen this virus. So hence the virus spreads very rapidly, because it is, imagine the analogy of a wildfire in a forest. So if the forest is full of dry trees, then the fire will immediately spread very rapidly because nothing is stopping them. Similarly here, the entire world population is susceptible because we don't have any pre-existing immunity. Hence the virus will spread very rapidly. And people who have certain underlying medical conditions or certain genetic composition, then they will end up getting more severely ill and perhaps would die. And the table here on the right hand side also gives a clue about the severity of the virus and the potential spread it could cause. So if you look at Ebola, Nipah, which is only recorded in 2 countries, Malaysia and India. And the reason is that this virus is spread by bats. So it only is present in those countries where this particular bat is inhabitant. So, but the mortality rate or the fatality rate is almost 77%. So which means that in every 3 individuals infected, 2 are likely to die. And Ebola, in every 2 individuals, 1 person is likely to die. But then now look at the 3 coronavirus epidemics happened in the 21st century. The SARS in 2002, and the MERS in 2012, and the current SARS coronavirus 2 in 2020 or 19. And these are some of the numbers. If you look at the MERS virus, which is the Middle Eastern Respiratory Syndrome virus, emerged in Saudi Arabia. And it was recorded that the mortality rate of this virus was almost over 30%. And if you look at the number of countries the virus spread is 28 countries, and the total number of global cases were just over 2,000. Contrast that with the SARS in 2002, it had slightly lower, but close to 10% mortality rate, spread to 29 countries, and recorded over 8,000 cases. And now the severity of COVID-19 is significantly lower than the other 2 coronaviruses. And look at the number of countries. So this number is much bigger now. It's over 200 countries the virus has spread. And the number I'll show you in a minute is now in millions. So apart from other reasons, one of the major reasons is that the more severe the virus is, in some ways it is much easier relatively to control, because you can spot who is ill, and then the quick isolation and treatment of those individuals will prevent the source of virus to the larger communities. But with the COVID-19 or the SARS coronavirus 2, the major challenge is that the virus is only relatively mild, meaning only 20% of people are likely to develop severe symptoms and perhaps need hospitalization. But the vast majority of the people infected, which is almost 80%, will not show any major clinical symptoms or perhaps very little symptoms. And there is also another challenge, is that the incubation period is relatively longer with this virus. So incubation period is the time when a person is exposed to the virus and to the time they start exhibiting any symptoms. So the problem with the longer incubation period is that if even some of us, if we are infected, we would not know until say almost like a week or 10 days. But then in the meantime, we'll go about our regular business, meet with friends or go to class, and you are shedding the virus, which is a source of the infection. And that is the reason why one of the recommendation is that we all have to wear masks, not because the mask is going to absolutely protect us, because the surgical masks or the cloth masks that we use may not fully protect against the virus. But the idea is that we all should behave as though we have the virus. So if everybody is responsible and wear masks, then the spread can be significantly reduced. And several studies have proven this. Wearing masks and social distancing has a significant role to play in preventing the spread of the virus, or at least minimizing the spread of the virus. So this was a snapshot of global cases I took on the 6th of March. You look, there are less than 100,000 or 98,000 global cases with over 3,000 deaths. And later in March, the cases were increased. And the next snapshot is taken in May. So now the cases globally, cumulative cases, are over 5 million, or 56 lakhs, with 3.5 lakh human deaths, 353,000 human deaths. And last night before I went to bed, I took a snapshot of the overall global cases, and look at this now. It is 8.4 million cases across the world, with over 4 lakh people have died. So, and if we are in a small town or a place, you may not see or you may not know many people that are either died, and we can come up with a lot of theories about, oh, this is not really that dangerous, or maybe India would not be affected as severely as Europe or US is affected. The question is it really true? And we don't have any scientific basis to believe that any particular country or a region is immune from this virus. So it's just a question of where we are in the curve of the disease. I'll show you in a minute what I mean by the stage in the curve. So before that, let us take a step back and a very quick introduction. Again, as I mentioned, a lot of this information was covered by Dr. Raghunatha Rao. I'll just try to fill in some gaps. So you all, I'm sure know by now, the name coronaviruses come from the Latin word corona, which means crown. As these viruses, when you look under electron microscope, they appear as though they're wearing a crown or a hat. And this is because they have these characteristic glycoproteins called as spike proteins on their surface, which gives the virus this appearance. So they're shown with these red pointy objects on the figure here. And another important aspect about these viruses is that these are largest among known RNA viruses. When I say largest, we're talking about the size of the genome. So the genomes are relatively, again, compared to other forms of life, they're very, very small. But within viruses, they're fairly larger RNA viruses. And the other important thing is, most of you perhaps know that in only viruses you see the genetic information is carried either in RNA or DNA. So all other forms of life, starting from bacteria, they have both RNA and DNA, whereas viruses will have RNA or DNA, but never both. So those viruses that have their genetic material as RNA, ribonucleic acid, they're generally more likely to be error prone, meaning when they replicate, they have to copy their genetic information. And RNA, when it is replicating, there is no what we call as a proofreading mechanism. So when DNA replicates, literally what is happening is that somebody is, if you imagine the copywriting notebook when we are in high school, right? So you have a sentence on the top, then you have to just copy the sentence multiple number of times. So the virus is trying to do the same thing. It has the genome that has these genetic code written in A, T, C, G, those nucleotides. And then it has to copy hundreds and hundreds of different times in order to make a lot of babies in the body, right? So when the DNA is doing that, there is always a possibility that it will incorrectly copy a letter. Instead of A, it might put a T here. And that is what we call as mutation. But in DNA, when this happens, there is an enzyme which will correct this, this mistake, which is called exonuclease activity, also called as proofreading mechanism. And this is not present in RNA. So inherently, RNA viruses are more error prone because nobody is checking their mistakes. So when RNA virus replicates, they constantly make these mutations or mistakes. And the more they replicate, and these mutations will accumulate. And I will also briefly mention about the antigenic shift and drift as was raised in the earlier presentation. So let us see where are these coronaviruses. Again, as I mentioned, we study these viruses. So based on the scientific understanding and estimates, it is believed that the coronaviruses have evolved and adapted themselves to live virtually in all species of animals, including humans, and perhaps over past 50,000 years or even older. So coronaviruses are not new. They have been there for a very, very long time, and they will continue to be there for the foreseeable future. And typically, they adapt in a particular host. So for example, coronaviruses in bats generally stay in bats and humans. We also have endemic human coronaviruses. As some of you might know, whenever we get cold, there are at least 4 different coronaviruses that just make us the common cold. So you just have a runny nose for a few days and you'll become fine. And most of us have had these endemic coronaviruses at some point or even regularly in our life. And the entire world population will have some kind of antibodies against these coronaviruses. And usually those coronaviruses, they stay in humans. And similarly, we know of coronaviruses in that affects chicken, or that affects cattle, that affects pigs. So all of those different viruses typically stay in their host, but occasionally they also jump species, which we call as interspecies transmission. And this happens because of, as I mentioned, as they replicate, they are making mistakes. And at some point, these mutations cumulatively will give the virus ability to transmit to a new host. And while this can happen randomly, what can enhance this transmission is the opportunity for the virus to jump. So for example, if the virus in bats is evolving and has gained the ability to infect humans, but if no people are adjacent to a bat, then that virus which has evolved or gained the ability to infect people has no opportunity to infect a man, because bats and humans are not close together. But in conditions and environments where such animal-human interactions are happening regularly, people are handling these animals for a number of different reasons. maybe for food or otherwise. And those conditions, which we call as hotspots for emergence of zoonotic pathogens, then at some point, the virus that has gained the ability in a bat or a different animal will now find a suitable host adjacent to the virus or the host. Then the interspecies transmission happens. And the challenge is that we would never be able to predict when and where this can happen. We can theoretically say this is a possibility, but it is absolutely impossible to precisely say when this can happen, because this process is completely random. And as I said, they evolve. And one of the important thing is that the proofreading mechanism, the lack of it, makes these viruses to be extremely vulnerable for mutations, and hence also have the ability to jump species. And the typical pattern of coronavirus emergence has two steps. The first one is coronaviruses in its natural host, most likely a bat or some rodents like rats and so on. And at some point, the virus that mutated and accumulated these mutations will gain the ability to infect a different host. And typically this host could be an animal, and which we call as an intermediate host. So in this first step is interspecies transmission. From a natural host, coronavirus will jump species and infect a domestic animal. And this is followed by a zoonotic transmission, because humans are likely to dwell closely with these animals, because animal farming or handling these animals for food generally puts or makes this transmission possible. And this is how the transmission of the previous epidemic viruses have happened. And somebody asked the question earlier, how do we know for sure that this COVID-19 virus or SARS coronavirus 2 has actually come from bats? And I'll just briefly talk about that in a minute. So this is the origin of other human coronaviruses that we know that happened in the past. So you can see that the natural host of most of them, if not all of them, are primarily bats. In a couple of occasions with the human endemic common cold type coronaviruses, they came from rodents. And they may be an intermediate host, they may not be. And the reason there is a question mark here is that A, we don't know if there was an intermediate host, and B, if there was, we haven't found that yet. So again, whatever we say today is based on the best possible scientific information. So the reason we say that this virus has come from bats is because we were able to find a very closely associated or related virus in bats. And that is the best possible theory. And has it been completely proven that this is exactly where the virus came from? Certainly no. But until we have an alternative theory that is much more stronger, the current scientific understanding is that the best possible explanation to this is the virus has potentially originated from bats. And the reason is that, again, this was touched upon earlier. Bats are very unique. And whenever we think about bats, most people might think they're just these flying, creepy animals. And perhaps a lot of people may not know that bats represent a fourth of the all known mammalian species. There are more than several thousand bat species across the world, and they are distributed the entire world except in Antarctica. And they're very, very unique because it is the only flying mammal, and they have a lot of important useful functions to play. They help in pollination and so on. But the way they have evolved, they have also evolved to coexist with viruses. So the viruses in bats generally do not make them sick. So Dr. Raghunatha has mentioned earlier, we know a whole host of viruses, for example, Ebola, Nipah. Most of the deadly viruses known to humans are potentially originated from bats, or bats serve as the natural reservoir. And this particular study has found that when they processed all these close to 20,000 samples from different animal species, they found almost 100 completely new coronaviruses that have not previously been described, and most of them are in bats. So bats do serve as the major coronavirus reservoir. And I quickly put this slide as I was listening to Dr. Raghunatha's talk about antigenic shift and drift. So this is a cartoon I made regarding influenza viruses. So let us try to understand these two concepts of how the viruses evolve. So in the first mechanism called as antigenic drift, which is a slow process by the name, a virus, typically an RNA virus, the example shown here is influenza virus, infects humans. And typically, a certain proportion of people will develop antibodies, either naturally infected or through vaccination. Then you have a sizable immune population. So what happens is this host immune pressure will select virus that will escape these antibodies. So gradually, the virus will undergo certain subtle changes on their surface glycoproteins to the extent that the existing antibodies in people are no longer able to completely neutralize the virus. And that's when you need to revise your vaccine. And that is the reason why the influenza vaccine or flu vaccine is made every year, and we have to constantly update because the virus gradually changes. But it will continue to be a human virus, infects people, but the only thing is it has slightly become different to the extent that the existing immune system or antibodies are no longer able to recognize. In contrast, the antigenic shift is a much more dramatic event where two viruses infect a host simultaneously, and then there is a possibility that they exchange the genetic material. And this will result in a completely new virus from both parents. And this resulting virus can gain a completely different ability that none of these viruses have. For example, the swine flu pandemic happened in 2009. We believe that the virus has evolved from, it has taken bits and pieces from avian viruses, swine viruses, as well as human viruses. And in theory, this genetic recombination or reassortment also happens in coronaviruses, but it is a very, very rare event. But when it does happen, it will lead to a dramatic change. And most often, since this is completely random, the virus has no brain. And many times these reassortments or mutations will end up being not very helpful for the virus. The virus may die. But very, very rarely, the combination that might emerge randomly can give the virus a completely different ability. So these are the two mechanisms that contribute to the evolution of viruses. And what we know about the SARS and the MERS is the story is very similar, although the context and the animals are different. So look at the top part of this picture, a virus that was circulating in bats for many, many years. Again, we believe that the virus has transmitted around early 2002 into an animal called palm civets. And humans, when they're interacting with these animals in a vet market, had exposed. So remember the earlier cartoon I showed you, the cross-species transmission, the first step, followed by the zoonotic transmission, second step. And similarly, fast forward 10 years, exactly same thing happens, but this time the players are different and the place is different. And this happened in Saudi Arabia, a virus that was circulating in bats over 30 years makes its way into camels, and humans that are interacting with camels then pick up the infection. So zoonotic transmission. And typically what happens is in both of these instances, someone is suffering from an illness that is, they don't know what is happening with them. They have some mild respiratory symptoms. And typically what they tend to do is, like even we do, go to a hospital. And before the pandemic or the epidemic is fully recognized, the medical professionals in the hospital, like doctors and nurses, will have no idea what this person might be bringing. So they'll try to treat the individual, and in the process, they may also have picked up the infection. So the person may or may not be hospitalized, but when these doctors and nurses go back home after treating this individual, then they will also spread the virus to the community. So initially, the concept is called as nosocomial transmission, meaning the virus spreads in hospital, followed by community transmission. And this has been recorded to have happened both in the SARS epidemic and the MERS epidemic. And which is very similar. That's why we call the health care workers are at a very high risk of disease transmission. And it's absolutely important that they have the PPE that they need to protect themselves against the virus. So again, the question was raised, do we know for sure is the virus came from bats? Again, as I mentioned earlier, this is the best possible explanation for now. And again, is it completely proven? Certainly not. So based on the genetic analysis that I showed you here, there was a virus in bats called as bat COVID-RATG13, which was found to have more than 96% sequence similarity with the COVID-19 virus. And based on that, it is believed that this is the parent virus that eventually gave birth to the current SARS coronavirus 2. And this virus is found in a type of bats called intermediate horseshoe bats. And these bats inhabit in this region of Asia, which is marked with red here. So these countries include India, Bangladesh, China, Nepal, Myanmar, and Thailand, Vietnam, and so on. And then, as I mentioned, the intermediate host, if you think about the interspecies transmission followed by zoonotic transmission, and the scientists have isolated a virus that was found to be similar or closely related to the SARS coronavirus 2 in animals called as Malayan pangolins. And based on this information, it is believed, again, if you look at the analysis, they align very closely with the SARS coronavirus 2 group. And based on that information, it is believed that perhaps these animals may have played the role as intermediate host. Again, at this stage, this is a brand new virus. We're still learning a lot of things. So this is, again, this particular theory about pangolins being intermediate host is also not fully accepted, and people do debate about this. But this is the best possible information. Again, literally tomorrow, this can change. If a different group have found a much more close related virus in a different animal, then we would have to revise our theory. And that is how the science work. So as of now, this is what we know, which can change in the course of time. So again, this particular point, I really want to emphasize that you may have heard that India will not be affected as severely as the other countries have affected. Is it really true? Is there any indication that any particular geography or people or country is immune or resistant to the virus? And at least based on the scientific information, there doesn't seem to be any, apart from some observational anecdotal studies. There is no concrete evidence to show any particular country or ethnic group or geography is resistant to the virus. And this is a cartoon that was made by the Zurich University in almost 10 years ago, 2010. And what you will see in a minute is that all these tiny little dots represent flights that move from one part of the world to the other. And there is also a black shadow that moves, which represents day and night. And this is 72 hours of air travel condensed into a few seconds. Just take a look. So see the moving dots, and the bulk of the flights from China, they touch Europe as well as North America. And there is then second level of air traffic to the South America, Africa, and countries like India. So now keep this in mind and look at this next global spread. So I captured this video until May, and starting from the 23rd of January, and this is the global spread of SARS coronavirus 2. So on the 23rd of January 2020, we know the virus was found in China. So China map is colored with a darker color. And as the countries go darker, it indicates that the virus has spread in those countries. So take a look. So the timeline spreads from early January, and once you start hitting into February, the virus makes its way into Europe and then to America. So I'll stop here. At this stage, look, none of these countries, either India, South America, or Africa, have the virus. So the virus has initially spread to Europe and Britain as well as North America, and simply because the most people from the infected parts have flown, have arrived in these places, especially in cities like New York and major international hubs. And that's what the transmission happens. And if you follow the timeline again, as the time progresses, as more and more people subsequently travel, then the other parts of the world will start becoming brown. So the point is that the transmission pattern reflects the global air traffic and the global movement of people. And it is just a matter of time when we got the virus. If you got the virus early on, then you're already increased transmission in the community and you are ahead of the curve. And those countries that got the virus later on, they are still catching up. So the key point is, unless we know the prevalence of the virus, which is only found out by extensive testing of people, which is not happening in any part of the world, we do not simply know how many people are infected in India or even in the US. So it is completely an unknown. And we only have been documenting severely ill, hospitalized people, as well as people who die from the virus. So that is the important thing to keep in mind. And as I mentioned here, the severity makes the virus difficult to control. And some people will ask you, why is this virus is different? And how come the SARS and the MERS just died down very soon and didn't really cause a big pandemic? And why COVID-19 is causing it? And there are all sorts of conspiracy theories. And I also saw some videos claiming that World Health Organization has been fooling the world, and COVID-19 is not actually a virus, it's a bacterium, and all sorts of theories. So remember, when people are paying attention, and there is also information that is spread, which is probably not based on facts. So we need to be really careful about understanding. And there is also a theory that somebody has proposed that, oh, this virus was manufactured in a lab. It has some parts of HIV virus and some parts of this virus and that virus. And all of those things, I can tell you, are not based on any scientific facts, because we study these viruses and we know all of those are completely baseless. And the reason is that the disease is less severe. And since most people, or many people, are asymptomatically infected, and that is contributing to the spread of the virus. So again, I heard earlier a question about in some cities there are more cases, in some places there are less cases, and is it because of the temperature? And I think based on what we know, the answer is no, because look at this global temperature map. And the temperature across the world is not the same. So when it was very cold in the US, the virus was spreading. And at the time, India was fairly warm, as well as Australia, and the virus was spreading there as well. So the seasonality of the virus that happens with some of the endemic viruses, where the virus spreads like waves, and a wave will be in winter followed by a less transmission. And that will not happen with an epidemic virus, because as I mentioned earlier, the entire world population is immunologically naive, meaning susceptible. So at least we do not see a major contribution by the temperature. So the key is how closely people are together. If people are close, even if it is extremely hot, you will get the virus. So temperature has no role to play as long as people are not social distancing themselves. If people are close by, you congregate, you will end up picking up the virus. So then the question, how does the virus spread? Again, this we will not be able to see. This is really critical. And even if you're not coughing or sneezing, when I'm talking, even if I'm not talking, I'm constantly breathing air out. And in the breath that I take out, there are tiny little droplets that we cannot see with the eye, which are currently, as I'm talking now, I'm releasing these small little droplets that are spraying around the area. And if somebody sneezes very hard, and the studies now show that the droplets can travel as far as 9 feet and 6 to 9 feet. And the social distancing is based on the fact that if somebody in your vicinity is infected with the sneeze, then are you in the area of their sneeze zone, so to speak? So look at this animation. So when somebody sneezes, this is what is happening. But the challenge is you can't really see these things. This was done in a lab so that those droplets can be visualized. And these droplets have Can somebody mute their microphone, whoever it has it on? So these droplets carry the virus and they deposit on surfaces. And what happens then is the virus could, and I also saw a question from earlier presentation in the chat, somebody asked, when the virus cannot survive in the outside the body, how come the virus is spreading? So the virus will not be able to spread outside the human body for a long time, which means that the virus does survive for a considerable length of time. And if we happen to touch the surface that has the virus during that time, and then we unintentionally touch our face, and we might be, putting our fingers in our mouth or rubbing our eyes, and this is how we end up infecting ourselves, and that is what happens. And hence the hand washing creates an excellent barrier, since you do not know where we are touching, especially when you go out. If you constantly sanitize your hands, then you are significantly reducing the transmission of the virus through these droplets on surfaces and we touching them. And that is an important thing that we need to remember. So I'll skip that. This one study, again, I have to caution that this is not the concrete information. We need to know more, but at least this study, which simulated conditions in the lab, has found that the virus does survive considerable length of time on different surfaces. For example, this study showed the virus remains live and infectious on plastic for 3 days and on stainless steel for 2 days. And they also compared with the SARS coronavirus 1 and how long it stays on copper and cardboard. Although these numbers might be a little different given the condition. So if you are outside a hot place in Rajahmundry, things might be different from where I am in Pennsylvania. But the point is that while the environmental factors do play a certain role, based on this study, what we can safely assume is that the virus does survive for a considerable length of time outside. Hence, the infection control precautions, such as washing your hands and not touching anything, or avoiding public places or avoiding crowds, absolutely makes sense from an infection control spread standpoint. So now the important point is about, and I also saw questions about why scientists are not developing a vaccine. Can we treat with ribosomes and all sorts of different things? So let us first understand what is happening in the body when the virus hits our body, right? So these are the key symptoms. So the virus is taken in, it is a respiratory virus, so it is affecting your respiratory tract. The virus initially replicates in the cells of the upper airway, the nasal cavity, and then it gradually goes to the lower airway, and you start developing symptoms like sore throat. This correlates with the virus replication in the lower airway. And that is followed by the virus, in some people, go to the lower part of respiratory tract into the lungs. And that's when the individual is developing the so-called dyspnea, the shortness of breath. And these are the typical symptoms: the fever, cough, and shortness of breath. And remember, all of these symptoms are because your body has recognized that something foreign has come into your body and it is trying to invade us. So your body is fighting back. And these are the symptoms caused by the inflammation, which is your body's reaction to the virus. The virus has completely no ability to cause any of these things. And these are the body's response to the virus. And that is something to keep in mind. So when it comes to treating, so what are we treating? Are we treating the virus, or are we treating the symptoms? And that is the difference in how do we talk about the therapy or the vaccines to this disease. So let us understand what happens, the pathogenesis of SARS coronavirus. So again, this is something that Dr. Raghunath Rao has touched upon. I'll just quickly go over it. So the virus enters into your body and replicates. And with any virus, same thing should also happen with the vaccine, is that we have, in most people that have functional immune response, we have a mechanism that will observe and identify that something that does not belong to us has entered in our body. And that is called as antigen. This is done by T lymphocytes. Recognizes that there's something foreign has come into the body, then it triggers a series of events in our body that leads into what is called as antigen presentation, followed by two arms of immune response, the cellular immunity as well as humoral immune response that makes antibodies. And along this time, these cells, the immunological cells, are also producing a lot of chemicals called as cytokines. And these are proteins, and they are meant to protect our body, right? And those chemicals will create these symptoms. So those chemicals, when they're secreted, they are causing the rise of temperature, and you're coughing or you're releasing this secretions and so on. They're all because of these chemicals. So since this is a new virus, and in some people who are immunologically compromised or have underlying conditions, or in some genetic compositions, the human body sometimes, especially with the new virus, they don't know how much to react. So basically, you have the analogy is that you tend to overreact to this. So the body, when it overreacts, these cytokines are excessively secreted, which is called as cytokine storm, and those cytokines are actually damaging your lungs, and that is called immune damage. So the virus is the trigger, but your body is almost like self-inflicting the wound. So we need to understand that even if you stopped virus at this stage, the body has already damaged itself enough. So at this stage, even if you treat the virus, it's not really going to help the people, because we have to now treat the symptoms, because the lungs are now being filled with fluid and they can't breathe. They need to be hospitalized and they need ventilation. So we have to understand, when you talk about therapy, what are we trying to treat and when are we trying to treat? So that will change what kind of therapeutic interventions that are needed. And that is something to keep in mind. There cannot be a one size fits all. So you take this pill, you'll be fine. I mean, that may not be the case with these rapidly replicating RNA viruses. And I also want to mention about the natural therapies and remedies. So again, I'm part of a bigger group here called Center for Infectious Disease Dynamics. We have several scientists that work with vitamins and mineral supplements and selenium and all sorts of different things and their impact on immune system. So we have to keep in mind that all those home remedies and the vitamins and the minerals, they're really good, and they generally keep our overall immune status better. So it's time tested, as Dr. Raghunath Rao has mentioned, and there is no dispute about that. However, when you talk about a rapidly replicating RNA virus like SARS coronavirus 2, even if you had a functional immune response, that is not really going to protect you, because what is causing the problem is your immune system itself. So the theory that the natural remedies alone can protect or treat the disease is probably not completely proven. And because of the fact that the immune system is reacting to that. So that is something to keep in mind. And here, this is what happens if somebody is infected. So it may not happen to all people, but some proportion of people will have this kind of progression. So the first day when the clinical symptoms start, the X-ray on the top, you see a small opacity on the right upper lobe, which is not, the entire lungs are finely clear. The person is breathing normally. And within a week, look at the lungs now, they're completely filled with fluid. And at this stage, the person has moved into a stage, what we call as acute respiratory distress syndrome. So the entire lungs were completely filled with fluid because of, as I mentioned, the excessive production of body's own chemicals that are trying to fight back the virus, but they're actually causing problem to your own lungs. And this is the time they need mechanical intervention. And at this stage, the antivirals are perhaps not of a great use. They will certainly stop the disease further progression, but they also need a lot of supportive therapy in the form of invasive mechanical ventilation, because they can't breathe. They need to be put on a breathing machine, essentially. And also another thing is, to resolve these symptoms, these individuals need to be given steroids or other anti-inflammatory drugs. And in this particular case, this was a healthcare worker, 31 years old, very young. And with the proper treatment and mechanical ventilation, this individual, a man, by the 36th day, remember, from the day of symptoms, it took almost more a month for this individual to go back home with the results of steroids and other. But for many people, that you saw, the graph I showed you, those hundreds and thousands of people, they're not a mere number, they're all real human beings. And what is happening with them is, unfortunately, they are not able to come out of this stage. So this is not the ending for many people, like happened here. In our Telugu films, there's always a happy ending, but with the virus, this may not be the case for many people. So even despite the treatment and mechanical ventilation, people may not make it. So that is something to keep in mind. And there are questions about whether women are at a higher risk, or what happens. Again, still we need to see a lot of data with the SARS-CoV-2, and I have interacted with a colleague of mine from South Carolina University, where they're finding that there may be vertical transmission, meaning when mom is infected, they also found the newborn baby was also positive. Again, it's an early data, but what we do know from the previous SARS coronavirus is that especially in the early trimester of pregnancy, there could be pregnancy losses or birth defects. And these are to a large extent attributed to the high temperature. So it is important that taking antipyretics to reduce the fever are critical, especially in pregnant women that are infected with the virus. So now quickly look at the replication of SARS coronavirus. Then it will be easy for us to understand how to stop the virus or where to treat. So very briefly, the virus attached to surface receptors, these are called ACE2, and it goes inside the cell and it uncoats itself. And this is an important aspect in the endosome. The virus needs to uncoat, and then the viral nucleic acids will then replicate, and then they go through the Golgi apparatus where the viral proteins need to be post-translationally glycosylated, and then the virus comes out. So the therapeutic targets of SARS coronavirus 2 try to attack many of these different steps. So for example, the antibodies or the convalescent plasma, the monoclonal antibody therapy also, they will prevent the attachment of the virus to receptors, the first step in the virus infection. So the antibodies will stop this step so that the virus is not able to infect. And the other aspects, for example, the hydroxychloroquine and chloroquine, what we know is after the virus infects a cell and goes inside the endosome, it needs to come out of the endosome, and they will stop that step. It is known that the hydroxychloroquine and chloroquine will prevent the release of the virus from the endosomes. And the remdesivir, which was talked about, is again a promising antiviral drug. It attacks the viral replicase enzyme called as RNA-dependent RNA polymerase, which the virus needs to make more copies of its nucleic acid. And when it is stopped, then you are essentially stopping or reducing the virus replication. And these are some of the steps. And the other aspect is that the viral proteins will go through endoplasmic reticulum, and they undergo post-translational modification. And for that, they require a certain group of enzymes. And currently, my lab is developing a therapy that targets this particular step to stop the virus replication. And this is, again, a tested strategy, which was also shown to be effective against other enveloped viruses. And we have also published a study. I didn't have the slide here. We published a similar therapy against Zika virus, and currently we are working on this therapy using blocking the virus replication or post-translational modification in the endoplasmic reticulum. So the question that was asked earlier is, when will be a vaccine or antiviral drug be available? So there are several vaccines and antiviral drugs are tested in the labs, and they're proven to be working, at least in the in vitro cell culture or in animal models. But the challenge is that the regulations in different countries are different, of course. But before we believe that a vaccine or an antiviral therapy is working, we have to prove beyond doubt that this therapeutic intervention is actually effective. And Dr. Raghunath Rao has mentioned about the different stages, preclinical, clinical stage of development of these therapies. So the preclinical is just a proof of concept. So we know that's the first indication that it is working. And there is no guarantee that what we found in animals or in cells in the lab necessarily translates into humans, because the biology is different. So the different stages of clinical trials are essentially, the first stage is to make sure it is not causing any side effects. So at the stage 1, we are not really talking about its efficacy, but we are talking about by giving these medicines is something happening to these individuals. So that is the safety part of it. And after the safety is ensured, then you go to the actual efficacy part, where the double-blinded randomized clinical trials need to be done, where you have to follow these individuals in a big cohort and treat them and then document all the data. And the reason that all these things need to be done is that in the process of trying to solve a problem, here the problem is COVID-19 and the solution we're trying to produce is a vaccine or antiviral drug, you don't want the solution to be a problem again. So then you'll end up with two problems. And that's why we have very tight regulations to make sure what we propose for any particular virus are effective, and hence they have to go through. And unfortunately, there is no way to fast forward this process. You still have to go through the clinical trials in people, and that is a frustratingly time-taking process. And that's why when we say the reality to people, it is not as exciting, because I might say, oh, the vaccine is not going to be available until the end of this year or early next year, as opposed to somebody saying, oh, now Italy has found a vaccine. This forms the headline in the news, and everybody shares it in their WhatsApp because it is a feel-good news that we want to share. But the reality is that it is not often as dramatic as they sometimes project it to be. So the reality is that it does take time, and these things are time-taking processes. But the lot of drugs have currently tried, the remdesivir and chloroquine, and there are some reports, especially chloroquine was found to have some adverse side effects in a small proportion of people. And in the US, the Food and Drug Administration has officially stopped all clinical trials with hydroxychloroquine. It's no longer being allowed here. And this is the discovery stage. So under normal circumstances, look at this, the drug discovery in the preclinical takes around 3 to 6 years, and the clinical trials will take anywhere between 6 to 7 years. And then the scale up, after we find the drug and it was proven to be effective, in order to make it available to the entire world population or a population of a country, you need to manufacture in a very high scale and packaged and made available. And that scale up takes at least 2 years. So currently, since we are in the face of a pandemic, this process has been cut down, because we are not going through a novel drug discovery, but we are trying to repurpose drug. So we are already finding a previously proven drug, and we are finding a new use for the drug, like chloroquine and so on. And that will shorten this process. And then, since most of them have a certain safety profile, you can quickly go to the phase 1. But the phase 2 clinical trial will still has to happen, and that will take several months, even if we. And what companies are doing now, they're taking a very big gamble, rather than waiting for the clinical trials to complete and to scale up, which will significantly delay. They're simultaneously manufacturing these compounds so that these two are overlaid. So, which is a gamble in the sense that if the phase 2 proves to be not successful, then they probably have bulked up millions of dollars worth of drugs simply to go in the drain. But with regards to this global pandemic, companies are making that huge risk with the hope that when they are successful, immediately the scale up has also happened simultaneously, so the widespread availability can be implemented. I will skip this about the antibody. So maybe I'll quickly show what is happening with the antibody response. So the virus infects the lower airway, and there are two types of antibodies, IgG, IgM. We know now that they are simultaneously raised, and the IgG perhaps will last for 1 to 2 years. Again, we have to cross-check this, and we have done some studies recently that shows many people who are exposed are indeed developing antibodies. So Dr. Raghunath Rao has mentioned what kind of antibodies are these. So we need to also make sure that these antibodies are functionally relevant, which requires a particular assay called virus neutralization assay. I'll show you some data in 1 second. This is the study that we published with a lot of people. This is the first ever plasma transfusion study in the US, several hospitals involved. And this is a preprint currently under consultation with the journal. But the key idea is that we showed that the virus neutralization antibodies nicely correlate with the ELISA if you test them with the ectodomain or the receptor binding domain of spike protein. So many people that have 1350 and higher ELISA level, they also have a log2 VN titer of 160. which is currently believed to be protective. So a good news is that majority of people who are infected and recovered, they do have antibodies. And the questions about reinfection are based on what we know about viruses. The reinfection is probably because of the problems with the testing. So for example, if your test has slightly lower sensitivity and specificity, then there is always a possibility of false positives and false negatives. So we believe that the so-called claims about reinfections are perhaps due to errors in the testing and not necessarily that the people are reinfected again. Again, these are evolving situations. We have to learn more. So in the meantime, what we can do is that the recovered people have these virus neutralizing antibodies, and once you test them, and the serum can be taken from them that contains antibodies, and that serum can be injected to either high-risk people before the virus exposure as a prophylaxis, especially if you think about doctors and nurses, or somebody who is infected, they can be given this plasma as a therapy. And based on currently Mayo Clinic in the US is doing a fairly large clinical trial with the convalescent plasma therapy, and so far 20,000 people have been transfused, and we are seeing a pretty encouraging data that this is safe. And there is also early indications that there are benefits of the plasma therapy. Again, this is still in the next few days, we will know more about this. So this is one thing we can do. In the meantime, vaccines or antivirals are not available. But the other important, very, very positive news that I would like to share with you, I don't know if some of you have seen this or not, that a drug which is very, very easily available across the world has now been found to be very effective to mitigate the serious illness due to COVID. So this is called Dexamethasone. And if you remember the earlier slide I showed you, the severe symptoms in the lungs is because of inflammation. So the anti-inflammatory drugs are critical in resolving those symptoms. And this is a steroidal anti-inflammatory drug. And in one of the world's largest randomized controlled clinical trial done in the UK, they have enrolled around 2,100 participants and received this drug. And when they compared with around 4,300 people, these people have significantly performed better. So this is a very, very great news. It just came a couple of days ago. And I think this is going to significantly help in minimizing the number of human deaths, especially in high-risk population. And in the meantime, what we can do is important is social distancing. I cannot emphasize enough. And there is always this question about when we can go back to school or when life can be normal. We need to remember that this is one of those defining moments in our history, in our lifetime. And perhaps when you become old, especially those who are young, you might tell this story as an interesting story to your grandchildren, because this happened when I was like whatever. And people in the 1918 who lived through that pandemic have a lot of stories to tell about this. So clearly the pandemic has changed the way the world is. And in the next 2 years, the world is not going to be the way it was maybe 5 months back. So we need to understand the new reality and try to cope with it. So the question is, we have to be responsible. You may have elderly people or young people in your family that could be more vulnerable, or there are people who are immunocompromised. So as a society, if we act responsibly, then we can protect our most vulnerable. And when the vaccine comes available, then it is going to significantly change, because vaccination will have protection. So then the final question is about the herd immunity, when it will happen. So the herd immunity, the concept is that if 60% of the 60 to 70% of the population has antibodies or immune, then the virus spread will minimize. But the question is, how do we get there? If we get a vaccine and then get a herd immunity, we'll get there in a much more effective way without losing a lot of human lives and not paying a lot of human cost. But if we, in the meantime, if we don't follow these precautions, then we will get there rather in a very, very negative way, because the virus will spread, and in the process it will infect people. We all will then end up getting herd immunity, but the human cost of this is going to be incredible and really devastating. So the key aspect is that all governments and organizations are mindful of this, and that's why these restrictions. And while we know we need to balance our social economic interests with the public health interests, and there are safe and effective ways of going back to business, but then doing things differently. So wear a mask, or do not go to a movie theater where people are crowded and sitting there for 3 hours. So things that can be avoided must be avoided, and keep the distance, and then wear a mask and wash your hands. These are the things that we need to do until at least early next year, when the vaccine becomes available. So that is so this is not going to end anytime sooner. And based on what we in the scientific world believe, is that since the virus has now spread to so many countries in the world, perhaps this virus is not going to go away like SARS and MERS. It will going to stay with us, and we need to learn to live with it. So meaning the virus may become endemic, and we may have a vaccine. So until we get a vaccine that is widespread available and implemented, we as the world and as humanity need to continue to follow these precautions. Otherwise, the graph I showed you will just become a number. That number will continue to increase, and hundreds and hundreds and thousands of people dying across the world. And every single citizen and every single individual have to feel responsible. Even if I think that I'm young and I'm very strong, I have to also be thinking about all my fellow citizens and be a good neighbor. So please wear a mask when you are in public places, and avoid crowds and gatherings. And that is the most effective thing that we can do in the short term. And also more important for this group is that please learn your facts, and do not believe whatever that might get shared in your WhatsApp. And if somebody said it, it doesn't necessarily become true. So you have to be very vigilant about fact finding, and be mindful about do your due diligence. If somebody says something, then you have to really ask the question, is information true? What is the basis for this information? So you have to be careful about the conclusions and believing the information. So remember the facts, follow the science, and then take precautions. Those are the important things. And with that, I will end my presentation, and I'll be happy to answer any questions. And once again, thank you very much for your attention. You've been a wonderful audience. Thank you.
Thank you, sir, for this very informative lesson. And I can see many slides which are making everything visible. Yes, seeing is believing, sir. You are very right, and the presentation is very excellent, sir. Audience, participants, if anyone of you have any doubt regarding sir's presentation, you can raise your hand. Then you will be unmuted and you can ask your question. Otherwise, drop the question in Q&A box. I think we are running late, and
yeah, I might take one or two questions before I need to go to my next meeting. Looks like between the two presentations, most of the questions are answered, which is great. So again, if you have any further questions, feel free to email the questions to Dr. Deepthi, and I can respond to the best of my ability. Yeah. Okay.
Sir, little question, sir. Small question, sir. Sir, we came to know that some people, they remain as asymptotic carriers of this coronavirus, no, sir? So how long they remain as asymptotic carriers, and how do we know that such a person can be like separated, or they can be isolated in order to arrest this transmission?
Great question. So the first thing is asymptomatic transmission does happen with this virus. And again, we are still learning. So the asymptomatic transmission, or people can be asymptomatic, or it could be it could be even pre-symptomatic. So for example, if somebody is infected and they have not exhibited symptoms, there is a period in which they are what we call as pre-symptomatic. Or somebody will not develop any disease and they remain as asymptomatic. So typically, this could be a window of 3 to 6 days or up to 10 days. But the challenge is that there is no way we are going to find out unless a widespread testing is done, which is not practically feasible and currently is not being done across the world. So we literally have to go and test every single person. And that is the reason why the recommendation of wearing mask is not because it will protect us. It might to some extent, but any of us could be asymptomatically carrying, right? So we should all behave as though we have the virus. And that is the only way we can prevent the transmission. And the two words that I also wanted to mention is isolation and quarantine. The isolation is only relevant or useful when you have tested somebody and they found positive. But until such time, you're only quarantining them because you suspect that they may have the virus. So the terms are different. And the biggest bottleneck in the world is currently lack of resources for the testing. And hence, you can't really test anybody or everybody that needs a test or that doesn't need a test. But eventually, when this testing system becomes more prevalent and widespread community surveillance type testing need to be done, so we know how many people, for example, in Rajahmundry have actually got the virus. We have absolutely no clue about that currently. And that is the limitation.
Can't we go with the blood test, sir, to know about the corona positive? Only swab test can be.
Yeah, so I wanted to make it very clear about the two tests that are available, right? So the virus testing, which is done by RT-PCR, and then the serum testing for antibodies. And again, depending on how they are done, the tests have problems of sensitivity and specificity. If it is 95% sensitive, meaning there are 5% of people that are not being detected by the test, right? So the RT-PCR test will detect the virus. And if you remember the graph I showed you initially on 0 day, when somebody is infected, the virus levels go up and they will drop after the antibodies go up. So there is only a certain window in which we can detect somebody by the PCR. And followed by the antibody test is a signature that is found in the body. And so, for example, there are scenarios if somebody is antibody positive, which gives an indication that they are exposed, but it doesn't mean that they may have the virus. So if somebody was exposed like a month ago, and they will be the RT-PCR negative because there is no virus coming out of them, but they remain serologically positive because they have still antibodies that will last for maybe 1 year. So both tests have different applications. So the virus testing RT-PCR is important to understand the active infections so that we can prevent the transmission. But the antibody test will help us understand the population level epidemiology of the disease. So for example, if you want to know how many people in Rajahmundry have currently been exposed, and that is better done by antibody. So both tests have different applications and they are not mutually exclusive, if that answers your question.
Right, sir. Thank you very much, sir, for sparing your valuable time. And
sir, I have a question. Hello?
Sure, yes, go ahead.
Yes, ma'am.
Yeah, I'll just throw some light on the evolution of the virus and its virulence.
Yeah. Again, these are thank you, Dr. Ramaneshwari. These are very big topics, and I probably need a lot of time to explain, but I'll be very brief. So the evolution of the virus and the virulence are two separate aspects. So the virulence is how severe the virus is going to be. And if you remember the earlier slide I showed you, there are different viruses with different levels of virulence. For example, Nipah virus kills 77% of people, highly virulent. So is Ebola. This is less virulent. And the other question is about the virus evolution. As the virus replicates, the viruses are making these mutations, and that will gradually change the virus nature. So there is a theoretical possibility that when the virus transmits from people to people for many, many times, then these mutations will accumulate, and theoretically the virus can then become more virulent or less virulent. So again, until we see, there are claims about certain parts of the world has a more virulent virus than in certain other parts of the world. And currently we don't have a strong basis to believe that. But to answer your question, with any RNA virus, these things are theoretically possible. The virus evolution can make the virus more or less virulent. But this is something we cannot predict, and that is the reason why scientists like all of us across the world are constantly studying these viruses, so we know how the virus is evolving. And there are several thousands and thousands of viral genomes currently being recovered every day, and we constantly analyze them. But this is something that we have to watch and learn. And in most cases, there is also a possibility that the virus may become less virulent, because remember, in the virus's best interest, the virus should not want to kill the host, right? This is like almost the analogy of, we don't want to set our house on fire because I want the house to live in. So the virus, in its best interest, wants the host to live. But in the process, as the virus evolves, sometimes the virus will also learn to mellow down, or the human body is trying to learn to live with the virus. And that's what happens after some time. Then the virulence could potentially go down because the virus has learned and the bodies have learned to coexist together. But again, with this virus, we have to wait and watch to see what the situation would unfold into.
Thank you, sir.
Thank you.
Now I request
good evening, sir.
Lord Principal Ramaneshwari Madam.
Madam, question, ma'am.
Hello.
Hello.
Yeah.
I think one question.
Sir.
Yeah, go ahead, Dr. Jagan Mohan Reddy. And I just wanted to say I have another appointment. So after this question, I unfortunately have to leave. But certainly go ahead, Dr. Jagan Mohan Reddy.
This is Dr. Uday, sir, from Vikram Sampur University, Nellore, sir.
Sure.
Sir, being a RNA virus, why it is carrying endoribonuclease, sir? Is it having any evolutionary significance?
So you're talking about the RDRP, the ribonuclease enzyme?
Yes, sir. Endoribonuclease.
So we need to think about the replication of these RNA viruses, right? So there are positive and negative sense RNA viruses. So when I say positive sense RNA virus, the viral RNA genetic information, it can be used as messenger RNA when it is infecting the cells. In contrast, some viruses needs to still transcribe their RNA into messenger RNA, and those viruses are called as negative sense RNA viruses. So for example, influenza is a negative sense RNA virus, and whereas this is a positive sense RNA virus. So the replication strategy of these viruses also depend on what kind of genomes they carry, and accordingly the enzymes and the different components they package into the virus are in line with what the kind of genomes they have, if that answers your question. Again, if you look into the differences between negative sense and positive sense RNA viruses as families, you can probably get more idea of that. Dr. Uday, did that answer?
Yeah, thank you, sir.
Thank you.
Ramaneshwari Madam, are you there?
Yes.
Ma'am, please go ahead.
A very good evening to one and all. And it's my duty to thank the speakers of the day, Dr. Raghunatha sir and Professor Kuchipudi Suresh Verma sir, who have taken the time out to throw light on the pathophysiology of COVID-19 and the possible ending. They have given us extensive insights into the structural and the physiological and the pathogenesis of the virus, and what can be the hope that can possibly come up in soon seeing an end to it. And I think the word possible ending is the most optimistic word today, that after the 3-month lockdown period, we have locked down our university with the awareness program that we have created on the campus to the students and to the members of the teaching and the non-teaching members of the campus. And today, Deepthi has brought us to the point of possible ending, I think, which is the most optimistic. Though still the origin of the coronavirus is still in debate, whether it is man-made or whether it is a natural one. Yet this coronavirus has opened up studies in different areas, research frontiers, and especially the structural studies, the molecular, the genomic studies have helped in pushing through to find some remedial measures and to mitigate the effects of the coronavirus. And I think the therapeutics, before we can find a vaccine, the therapeutics, as Professor Suresh Verma sir has mentioned, that there are more than 200 drugs in the pipeline, and since and at different phases of development. But in the last few days, some words like dexamethasone, then remdesivir, then another word also I have come across, that is fluxergion, which these words have brought us some hope and faith that soon we are going to see the end of the coronavirus disease, and put us back onto the track so that we can have a peaceful and stay here. And I think as Professor Kuchpudi Suresh Verma sir has pointed out, the best possible way to stay safe is wearing masks, then social distancing, and at the and with utmost care, washing hands can help us to stay safe. And again, I once again thank both the speakers for giving us such insights into the COVID-19 disease. Thank you very much, sir. And I thank and congratulate Dr. Deepthi for organizing this webinar, which is very informative, and the participants have remained enthusiastic about this webinar throughout the time. And thank you very much, Deepthi.
Thank you, madam. Thank you. Sir, thank you very much, sir. I think you are busy.
Yeah, I need to move. But thank you very much again. I just want to echo what Dr. Ramaneshwari has just mentioned. So thank you for organizing this webinar. It's been a wonderful experience. And I also want to appreciate the university for taking the initiative. So thank you all the participants for your patient listening. And I see a lot of questions. I wish I had more time, but it shows the enthusiasm. So this is again maybe a trigger for people to learn more. So thank you all and have a wonderful day.
Sir, thank you, sir.
Deepthi, can we leave?
Madam.
Can we leave?
Yes, ma'am. Just conclusion చెప్పండి participants కి.
మీరు voting thanks చెప్పండి.
Oh, okay, ma'am. So, dear participants, thank you for your patient listening. I think we had a wonderful sessions by eminent speakers, and of course we could not pay much time because the speakers were busy and all the doubts could not be cleared by them. Still, I think that many aspects related to COVID-19 got cleared, and thank you for your support. We have a feedback link in the chat box. You're all requested to give your feedback through that link, and once you give the feedback, you will receive your certificate as soon as it is done. Thank you once again. Thank you all.
Deepthi ma'am.
Srinivas,
Ma'am, we don't see any link here, ma'am. The chat box.
మేడం వినిపించట్లేదండి. మేడం వినిపించట్లేదు. మ్యూట్లో ఉన్నారు మేడం.
నన్ను హలో సార్, కెన్ యూ హియర్ మీ?
మేడం నేను పద్మారావు.
Yes, Padmarao.
మేడం
Participants, please find the link dropped by Dr. CHV Padmarao at 6:28 p.m. in the chat box. At 6:28 p.m., Dr. CH Padmarao has dropped the link for feedback. I hope you all got it.
అటెండెన్స్ లో ఉన్న వాళ్ళందరూ ఫిల్ చేశారు మేడం ఆల్రెడీ.
అదే అంటే ఎవరో కనిపించట్లేదు లింక్ అని అన్నారు, అందుకని
పెట్టమంటారా మళ్ళీ?
ఒక్కసారి మళ్ళీ పెట్టి ఇంకా క్లోజ్ చేసేద్దాం. శ్రీనివాస్ గారు మీరు ఉన్నారా? హలో, Padmarao, can you please paste the link once again? Padmarao ఉన్నాడు. సార్ జగన్ సార్, హలో. సార్ జగన్ సార్,
మేడం క్లోజ్ చేయమంటారండి?
ఇంకా క్లోజ్ చేసేసాయి. పద్మారావు క్లోజ్ చేసేసాయి.